Welcome everybody, welcome to the stream. Hello, it's been a while since I've gotten to chat with all of you, and today we have a very fun and exciting stream. Welcome. Hello Gluon, Dreamless Sleep, welcome. Hello Clockwork, hello Astral, welcome Jareer Money, hello, welcome to this stream. As you can probably tell from the picture that I have up here, the flu video is in fact almost ready to go. It will release on Wednesday come hell or high water. I am just putting the finishing touches on it. It's been a long time coming, and I'm very excited to show you that when that is completed. No more rewrites. I'm happy with the way that things have gone, although I wish it just didn't take as long. Hello, welcome Lunkel, welcome. Today we are going to be talking about the prion protein, of course, a fascination of mine, and has been a fascination of mine since I was very young. But before we get into that, I just want to start off by talking a little bit about the prion protein as a whole. Before I do that, I actually want to point out that we do have a new glossary command. So this replaces the old jargon command, and I think is formatted in a lot in a smoother way. So instead of having a disordered list of all the definitions, you now can get at an actual glossary of terms that I will be using, so that you can just look up the individual terms as you need them. And I think that will make it a little more readable, I think. All right, with that being said, let's go ahead and get started. So if you are a viewer of my channel, there is a very low chance that you do not know what a prion protein is, but in case you do not, here is a brief description. Prion protein, specifically the major mammalian prion protein, is what is called a prion, which is an infectious membrane, an infectious protein. The mammalian prion protein specifically is a membrane-anchored protein with a difficult-to-determine function, right? So the prion protein, when it's happy and healthy, actually lives inside the membrane, and it's anchored in the membrane. Aggregation is behind some of the worst neurodegenerative diseases known to mankind. Of course, if you are a fan of biology, you do not need to be reminded, but just in case you do, the aggregation of this protein is implicated in diseases like fatal familial insomnia, Kuru, mad cow disease, CJD, etc. So these are all pretty horrifying diseases, very rare, thankfully, but if you want to sleep restfully at night, I would recommend not looking those up. So these proteins are surprisingly expressed in many different cell types. So you would think for such a potentially dangerous protein with no known function, or with a difficult to determine function, that the expression of the protein would be somewhat limited. But it's actually not. It's expressed in a lot of different cell types, and we'll actually talk about the implications of that down the line. Clockwork says, autophagy stream with no mTOR. Sounds like I'm leaving. I don't actually know a lot about autophagy. I learned just enough to be able to comprehend this paper, although maybe that'll be another deep dive later into the future. Okay, but before we get into the actual meat and bones, I want to address some of the comments that I got in my previous Prion paper video. Gosh, I can't speak today. So I had a couple of comments in my Prion video say that it was maybe asking for too much to find a function for the prion protein, because as the commenters have rightly discussed, evolution does not intelligently design proteins or organisms or limbs or organs, etc. So evolution doesn't actively sculpt proteins, right? There's no grand intelligence that we can prove, at least. And my framing of the prion video was that was surprising, because we have evolved the prion protein and kept the prion protein across a variety of different species. And so because evolution doesn't actively make things useful, people found that this was maybe assigning too much agency to the idea of evolution. I just wanted to address those comments. While evolution does not technically intelligently design species, sequence conservation across species implies that evolution was not free to play with mutating its sequence, right? And I want to pause here and define some of these terms before we kind of have an interesting conversation about what the implications of molecular conservation actually are, right? So what is sequence conservation? So up here I have an alignment between two different versions of the same protein. This is hexokinase. This is an enzyme that kicks off glycolysis, and you can imagine that something as essential as glycolysis, which is a metabolic pathway used to break down sugar into energy, that this would be pretty well conserved, and indeed hexokinase is pretty well conserved, meaning that this is a core protein shared by a lot of different species across all sorts of different domains. And we can see here that I have aligned the human version of hexokinase with the mouse hexokinase. And this is a very small segment of the protein, but we can see that there are regions of the protein that are conserved, right? These letters, oh, I can draw now, I forget. These letters, oh gosh, it is my mouse though, so it's not exactly pretty. These letters are the amino acid base codes. So the sequence starts valine, followed by, I think the R is arginine, although I'm forgetting. F, I think, is tryptophan. I don't really remember. But you can see that there is high amounts of agreement between the human version of this protein and the mouse version of the protein. But there are also regions of the protein where you have species-specific differences. This is actually the new format of BLAST, which is a little harder to read in my opinion. But you see here, everywhere where there's a plus in the middle of these two sequences, you have an amino acid substitution that is similar to the original amino acid. And if you see a blank, that means there's a complete mismatch. So these amino acids are quite different. I don't really know if I like this kind of formatting, but it is what it is. But so the idea of protein conservation is, if you look at the different versions of the proteins across species, they should vary depending on what species you are by a certain degree, usually correlated with how closely you are related in the evolutionary tree. F is phenylalanine. Thank you so much. What did I say? Tryptophan? Whoops. F phenylalanine. I should have known better. For some reason, I conflated that with PHE, which is the 3-letter code, which is different than a single letter code. Yeah, I'm gonna get I'm gonna eat crap for that, but yeah, thank you so much for the corrections. Yeah, so that's the idea of sequence conservation, that across evolutionary time it is normal and natural to expect that some proteins diverge from one another over the course of evolution. So conservation implies some sort of useful function, but this is not always the case, right? So this is actually a pretty nuanced idea. When you look at proteins like hexokinase, something that is so essential to the basic foundational processes of life, you would expect that once evolution figured out a way to do something that important, that it'll just keep on doing that without that many changes. And for the most part, that's kind of true, right? Like if you have an essential protein like in the cell cycle, those are really important. Things that are uniting in terms of our shared toolbox of life tend to be more conserved because they tend to be more important. But that's, again, not always the case, which I think is interesting. So why might a protein be conserved, right? Why might our hexokinase look the same as a mouse hexokinase minus a couple of base pairs? Well, one reason why a protein might be conserved is that there is some sort of phylogenetic relationship that is close, right? So we are closely related to gorillas, and so it makes sense that most of our proteins are going to look like gorilla proteins, right? A protein might be conserved, as we just said, if it has an essential function, right? If it's involved in glycolysis, the cell cycle, something that living things have been using for millions and millions and millions of years. That's probably not going to change all that much, because if you screw with it, you're likely to not end up alive. So those proteins are generally well conserved. And this is an interesting concept, although I don't know how often this happens, is that a protein might be evolutionary conserved if a lot of mutations in that protein end up with a deleterious gain of function, right? So this is more of a hypothetical, because I don't actually know of any protein that fits this definition. So this is basically saying a protein doesn't necessarily have to have an essential function, but maybe if you mutate it, that actually causes a problem instead of taking away something that you need. So that is, I think, a pretty interesting idea, although I don't know if that actually happens. So why might a protein not be conserved? Well, we can just take the inverse of the previous section. The phylogenetic relationship might be very far. So you would expect that if you compare human proteins with fruit fly proteins, that those sequences are going to look pretty different, or, different enough. A protein might not be conserved if it has a non-essential function, right? So if mutating a protein doesn't break the organism, natural selection has a lot more room to play with that protein. And so that might be a reason why a protein is not conserved. Or a third category, which is a very nuanced take, is that maybe it does have some sort of essential function, but is sequence-independent structural/functional conservation instead of sequence conservation, right? So proteins are strings of amino acids, and those amino acids will fold in a way that gives you a certain shape that's very useful, but you might not need those exact amino acids to form that same shape. So you might have conservation from a functional perspective, but not conservation from a information perspective, right? From an amino acid or gene level. So those are a couple of different ways that we can think about the nature of molecular conservation or protein conservation. So I want to kind of pause here and take any questions that we might have from chat. Actually, I'll probably end on that previous slide while I take a sip of my tea. So I want to just recap sort of the conversation that was happening in my comment section. This idea that the prion protein does not necessarily have to have a function because evolution is not actively sculpting proteins. There's no driving intelligence behind protein design. And so I am trying to have a conversation around that idea by talking about conservation. And the idea here is that we are going to talk about the level of conservation in the prion protein. But I want to kind of take a poll of chat briefly. Give me 1 second to actually get that set up so that I can sort of gauge the room. Because I think the upcoming conversation is actually going to be pretty interesting, and I want to make sure that we are all on the same page with regards to what I mean by protein sequence conservation. I should have had this prepared. So let's go ahead and open this, and I will go ahead and start a poll. So do we understand what I mean by protein what is going on? Don't know. Okay, start a poll. Do we understand what I mean by sequence conservation? And I've got the poll set up. I can explain it again if enough people want me to. This is making me think of someone I knew with mitochondrial disease. That's interesting. I wonder what that relationship is, Nato. Give me a sec to hydrate here. Get myself some orange tea, which is quite nice. I missed yapping about science too. I was very, very sad to not be able to stream last week, but this week we've got a monster of a stream. There's so many interesting findings with regarding the prion protein, and I feel like I only scratched the surface with that video. Even though at the time I thought I did a really comprehensive job, it's just the more I read, the more I realize I don't know, which is expected, we'll say. All right, it looks like, the people who decided to respond to the poll, we have a general understanding of what I mean by sequence conservation, so that's good. So I will continue right along. So how conserved is the prion protein? Well, I took a look and I did a couple of protein alignments, and this is what I found. So the left column here are different species, and on the right column we have percent identity with humans, so the amount of similar overlap that we have with different species. And so gorilla, our cousins, essentially, we have 99 sequence identity of the prion protein. That's pretty expected, right? We are, of course, very closely related to gorillas and all other primates, so that's not surprising. When we talk about mice, we are sort of similar to mice, right? They're not that far off of our evolutionary line. So 89% is still quite high. Cats are a bit further than us, 88. Camels are even further, 89. Pigs, 87. Elephants, 85. And then we have mammals that are very far related from us. Echidnas at 75, platypus at 58, and then chickens at 43. And so what's striking to me is that there's a pretty high level of prion protein conservation amongst mammals, at least mammals that we associate with modernity, right? It's because echidnas and platypuses, they're a little weird for mammals, a little, like the echidna is just old for mammals. Platypuses are just kind of weird in general, but I put them up there anyway because I found them to be interesting. But like from elephants up, these are what I consider to be like modern mammals, and there's a pretty high level of conservation there. Dang, the further you get away from other mammals, the more differences appear. Yeah, and that's to be expected. That part I'm not really surprised by. I'm more surprised that it's so tight from gorillas to elephants, right? This is a very tightly conserved mammalian protein, which I think is interesting. All right. What would we expect for this not to be the case? It's an excellent question, Astral, and so I tried to couch this in comparing to other examples. So I will get to that in just a second. But I first wanted to take a look at the tree of life, which you can do in the description for this stream. You can go to OneZoom and actually look at the tree of life. And here we have rodents and primates, right? Like rodents are more closely related to humans than all these other animals are. And then we have here elephants on this side, right? So from here, and then we have marsupials here. Echidnas are down here. So like the further we get away from where we are, the more variety we have. But everything like up here keeps the prion protein pretty tight. And then down here, things start to get kind of crazy. And Astral is right. This is not unusual. The longer we have to diverge, the more likely it is that we have proteins that diverge as well. So I wanted to compare it to some other proteins. So this is CDK2. This is a cell cycle protein of similar size, so we can sort of equalize some of those effects, and these are the percent identity with humans. So the gorilla CDK2 is actually only 88% identity with humans, and this is a very, very essential protein, right? This is something that is a part of this shared toolbox that all life has. Mice are similarly in that area. Cows, elephants are actually much closer to us in terms of their cell cycle proteins, and then chicken is down to 86, right? Like you should expect CDK2 to have high levels of conservation, but even here you see that there's some room for the evolutionary toolbox to play around with. So this is an example of a very well conserved protein, and I actually was unable to find proteins that are not very well conserved. It's actually kind of tricky to do so unless you get into like immune stuff, but immune stuff I always view kind of as special cases because every species has a very unique immune system because it has to co-evolve with viruses and bacteria and fungi. So I actually don't know any protein that it could have had for the opposite case except here. CDK2 more fundamental and like less than 300 residues, easier for higher deviation conservation. So the prion protein is of similar size, which is why I chose CDK2. So we can see a similarly sized protein with, we know, a very, very important role in life, right? Just to see how much that tracks. Although CDK2 is a lot older than the prion protein, right? It's got a lot more time to be established. And so to compare with another protein with less conservation, I chose to look at the Doppel protein. So this is, if you watched my Prion video, the doppelganger of the Prion gene. They share a common ancestor. It's likely that these arose from a gene duplication at some point during our evolution. And we actually see that, unlike the Prion protein, gorillas don't have Doppel. I think I tried looking for the sequence, and I think I found a paper that showed that for some reason gorillas just don't make this. And we know that Doppel is kind of important because Doppel plays a role in male fertility. So the Prion protein, we're still not super sure what it does, but the Doppel protein, we do have a better idea. It's involved in the acrosome, or the part of the sperm, making sure that it functions correctly. Macaques are down at 91, so already, even though this is another primate, right? Like that's a pretty big jump from 100 to 91. And then here you see cow, mouse, elephant, these other more closely related mammals, it's down to 76, as opposed to the prion protein, which is in the high 80s, right? So Alone, this doesn't really mean anything, and we've got hedgehog here, you know, for our distantly related mammalian cousins, but we can already see that even though this is a protein with a known important function, it is less conserved than the prion protein, and it has a similar age. That's really, really bizarre. I don't think silverbacks need to get any more manly than they already are. Yeah, maybe they decided to just jettison it from their genome. I don't know. Maybe they make Doppel. I just couldn't find it. It's really, really bizarre. But in any case, like these values here, right? 76, 72, 74, this is less conserved than the prion protein, whose function is kind of not fully clear. And we know from mice knockout studies that mice that lack the prion gene are largely normal, but they do actually have some neuronal defects, which I think is important to acknowledge. So Doppel is a known regulator of male fertility and has more sequence variation than its sibling, the prion protein. This, again, does not guarantee that prion has an important function, but for whatever reason, evolution was a lot more willing to play with the Doppel gene than the prion gene, despite being similar ages, although Doppel was lost in birds. So why might this be? The prion gene could have a cryptic function, maybe having nothing to do with the protein, right? There's all sorts of weird things that RNA can do, so maybe the protein RNA is what's actually conserved and the protein itself is kind of a side effect. Maybe it's got a redundant function, so if you remove the prion protein, maybe there's some other protein that picks up the slack, or a subtle phenotype, which might be behavior or memory or social behavior, something that's not easily studyable in mice, right? So we know that largely mice are normal when you knock the protein out, but you know, you can't interview a mouse, for example. And we also know that the zebrafish also makes its own version of the prion protein. This is a far ancient relative of the mammalian prion protein, and we know that in zebrafish the prion protein is actually important for memory. So this is not exactly the same protein. In fact, these proteins are quite different. So on top here we have the human prion protein up here in these lines, and then this bottom line here, that's the zebrafish. So you can see that there's a lot of blanks in the middle that indicate substitutions in the protein sequence that are not synonymous or not even remotely equivalent. And you can also see that the zebrafish sequence has deleted large chunks, right? This is a deletion over here, has deleted large chunks of the protein. So we can see that even though there's very little in common, there's still enough matches to confidently say that this is an ancestor of our current prion protein, and we know that this had a neuronal function before, which I think is also very interesting. So we know at some point, some version of this protein was pretty useful, in fact, very useful for the zebrafish. Okay, so I want to pause there because I'm about to get into the paper in general, and I just want to pause for questions. So I didn't fully address the question that was posed to me in my prion video of, hey, you're trying too hard to make it look like the prion has function, when in reality it could be vestigial or not have any function at all. And I think, you know, that's fair to say. And I think where that comes from might be the idea that we have vestigial limbs, not limbs, but like we have the remnants of a primate tail that is not super useful for us. You know, we have the appendix, which, you know, might have function, but if we remove it, we're largely fine, right? I think that's maybe a good comparison to make, right? The appendix probably had a function at some point and probably had a pretty important function at some point, but for whatever reason, the context deemed it not to be that important anymore, and we can remove it, no problem. So I think it's more fair to make that comparison as a potential possibility for why we still make this potentially dangerous protein, right? Because it's not completely right to say that nature makes no mistakes, that there's, you know, I'm not saying that the prion protein has to have a very useful function. I'm just saying that it's surprising that a protein that a lot of mammals make, that is pretty similar across most mammalian clades, it's surprising that we don't really know with great detail what it actually does in its healthy state. So I just wanted to open this conversation by addressing that aspect of the prion literature. Yeah. Any questions? I will have a sip of my tea, and if not, then I will keep on going. I do think that this is worth talking about because It made me think a lot, actually, about the role of conservation and protein function. Toes kind of belong to this category too. Typically not super useful, but the little toe frequently causes massive pain upon being stubbed on furniture. That is true. That is true. Trade-offs. We should all just have the little stick feet that I have. What am I looking at on the slide, by the way? Sorry, I arrived late. No worries, Leo. So this is what is called a protein alignment. You can do this by going to the BLAST function on the NIH NCBI webpage, which I have down a link below, where if you use that tool, you can actually copy and paste known protein sequences in the query and the subject lines and actually get an alignment, which is, so BLAST stands for Basic Local Alignment Test, I think. And so up here in the query, this is where you see the amino acid sequence for the human prion protein, and in the bottom, the subject, we have the amino acid sequence for the zebrafish here. So you can see that in the dashes, the zebrafish got rid of a lot of the prion protein, and then where you see blanks in the center, that is where the protein sequences don't align, where there is an amino acid substitution that is not synonymous, right? Like substituting glycine for tryptophan. Those are two very different amino acids, indicating molecular divergence. Wherever you see a plus sign, you see a substitution that is similar enough. I don't really like that. I wish they just wouldn't show the pluses, because that I don't think is very helpful for me. Yeah. What about the 2007 Westergaard paper that proposes some possible functions of the prion protein, including maintenance of oxidative stress, anti-apoptotic activity? I have not seen any additional literature come out of that paper. I haven't read that paper, although I do know that there are people thinking about prion proteins in the context of metal scavenging. But the idea is we, like, scientists have been able to show that the prion protein can do these things. It's a lot harder to really think about why we still keep it around. When we remove it, it doesn't really matter. And so this paper actually does not answer that question either. I just think it's kind of interesting. So I have not read that paper, Zach. I'm curious to see if we're talking about the same thing with regards to maintenance of oxidative stress, because metal ion stuff can overlap with that, we'll say. It makes sense that conservation can be an inconsequential mistake if changes can also be inconsequential mistakes. Yeah, exactly. So, wait, let me reread that comment. It makes sense that conservation can be an inconsequential mistake if changes can also be inconsequential mistakes. Maybe, but the thing about conservation is that you're battling entropy, because DNA polymerase, when you copy over your genome, that polymerase is going to make mistakes, and it actually takes energy to correct those mistakes. And so there's a molecular trade-off of the energy required to repair a gene and just letting the mutation ride. And a high level of conservation implies that mutations in that particular gene were not very well tolerated. Will it be possible for Homo sapiens to acquire extra limbs, for example? I mean, possible in biology means anything, because a lot of things are technically possible. It would just take a very, very, very long time and have some sort of selective advantage. The fact that we live so comfortably these days means that I don't think, as long as we have our modern amenities, that we'll ever see it happen. Although in the apocalypse, maybe. Could the conservation be a function of its context in the genome rather than a specific function per se? Yes, that is, I have that here in terms of its cryptic. Let me see which slide I have it on. I have it somewhere. Sorry for flipping through the slides, but I want to address that because I get to talk about that. Yeah. Deleterious gain of function. I swear I have that somewhere. Maybe it's on the next slide. Oh, cryptic function here. Yeah. So maybe having nothing to do with a protein. So the prion gene, I'm specifically using the word gene because I'm trying to capture your nuanced idea here that maybe it's not actually the protein that we care about, but there's something about the DNA that's important, which, you know, could be the case. In a very funny, cryptic way. Like maybe the prion gene is involved in a local genomic architecture that puts an enhancer gene the correct bases away from a downstream gene that it affects. Like there's DNA level things that the prion gene could be doing that would not be explained by its protein function, which I think is too much to get into for this stream, but I do really love that idea. I remember a paper where removed prion gene leads to some difference in hyperpolarization. Yeah, so neuron stuff, right? Like, it's probably pretty important for neuron stuff. It's just a mystery why the mice don't really act that different, but it is very interesting. These are different bases to GCAT, right? Yeah, GCAT are bases for DNA, and then these bases are amino acids, all right? So alanine, glycine, leucine, you know, a lot of enes go in here, the building blocks for protein. Yeah, tons of great questions. If I may ask, what topics are you guys talking about? We are going to be talking now about the role of prion proteins in skin cancer. All right, so excellent questions. I loved talking about that. I was so excited to share that particular piece just because I don't often get to talk about, you know, very nuanced stuff that is at the same time pretty accessible, I think. All right, so here is my content warning. We are talking about cancer, and so a part of this paper has to do with real data that captures the life of real people. And so, you know, just as a warning, you will see data that reflects people dying, which is of course unpleasant. If you watched my 12-hour glioblastoma stream, you know how difficult it is to actually read data when you know that the real, like, there are like real lives attached to some of these numbers. So just be warned that that will come up. Okay, so what is melanoma, right? So we're talking about the prion protein, and we're talking about its role in skin cancer. And so this is the particular type of skin cancer that we are talking about, the melanoma. So this is a cancer of the pigment producing melanocytes. This is the most dangerous type of skin cancer, and so because it's so dangerous, it's not a bad idea to occasionally check your moles, right? You know, look at yourself in a mirror, and if you see a black patch, a darkly colored patch of skin, it's a good idea to have that checked out, right? It's also not a bad idea to wear sunscreen when the UV index is greater than 3, right? Like, this is a cancer you do not want. Its only saving grace is that you have some control over seeing it coming, right? And so I'm not like a Saiken or Shiripan or Rico. I'm not like a healthcare professional, right? So I can't really tell you exactly if you have skin cancer, but just know that there's a helpful guide that you can find on Wikipedia. That's the A through G's of skin cancer. And so, of course, get your skin checked by an actual professional, but as a hedge against that, you might want to learn the signs of melanoma. So if you have a patch of skin that has asymmetry, borders that are irregular, you have a color that is there, diameter is big, and it changes over time, yeah, go check that out. All right, so I was really, really shocked at the fact that the prion protein was found to be important in the development of skin cancer. This was really, really shocking to me. Why is it shocking to me? Because, as you know from my prion video, I focused almost entirely on the role of prions in the brain, right? We know prions are important for neurodegenerative diseases. We know that they are expressed in the brain. So here on the left column, right, this column and this column, we see the amount of RNA in that particular organ. And you can see that there's a ton of prion RNA in the brain, some in the eye, which is, you know, the eye is just an extension of the nervous system, so I'm not tremendously surprised about that. The gastrointestinal tract, and then muscle tissue, skin. Like, the prion protein is made everywhere. That's actually really surprising to me, right? Because we only know, or at least I at the time only knew about its function in the brain. And on the right, we see protein. So it's important to note that protein does not necessarily line up with the amount of RNA, right? Protein expression can be regulated at a couple of different levels, including DNA level, including RNA level, and including protein level and after protein level. And you can see that in the brain, there's a lot of prion protein, but not a lot in the eye, even though it has a lot of prion mRNA. What I'm surprised by is that the second most populous region for the prion protein is in the skin, and that's really shocking. Really, really shocking. Yeah. Can we just use sunscreen to avoid most of the skin cancer? Using sunscreen will help. We'll put it that way. Sunscreen is great, and also keeps your skin looking young. Yeah. Yeah. Well, the skin and brain both come from the ectoderm. That does make sense, evolutionarily speaking, but I'm kind of surprised because, okay, so if we go with that, Lunkel, that's an excellent point. What else comes from the ectoderm? I'm not like a great dev bio person, but let's see if that pattern holds across all of the ectoderm tissue. So if someone could quickly look up what other terminal features come from the ectoderm, that would be really helpful in this case, because I'm really surprised that it's just the skin and the brain that make a ton of this protein. But that is a very interesting question. That one paper from the FiStream showing prion protein having tenuous immune system function. Yeah, that's really surprising too, right? Like the well, I had to take it down because I didn't have permission to actually show that paper. But yeah, the prion protein plays a role in the virology of the cytomegalovirus. I believe it's a cytomegalovirus, right? Something in that family at least, which is very surprising. Like it's coming up in all these unexpected places while we know of it mostly in the disease context. Okay. It reminds me of some neurotransmitters called melanocortins, who are also expressed in the skin. They're responsible for melanin production. Interesting. So there is a good bit of overlap then. That's really surprising. Okay, so this surprised me. It might not surprise you, but it surprised me. Okay, so the idea of a non-neuronal function for the prion protein isn't actually new. In fact, there are other papers showing here that the prion protein, and we're talking about the prion protein, the mammalian prion protein, is actually important for repopulating hematopoietic stem cells, which is really, really surprising, right? So it's not like we don't know anything about how the prion protein functions. It's always just a little surprising when we remove it that it doesn't appear to matter. But we do know that it does do things. And here we actually have another citation from the prion queen, Susan Lindquist, unfortunately passed away relatively recently. If you ever find yourself reading the prion literature, especially the yeast prion literature, you find her name everywhere. She is the prion goat, honestly. It's actually so crazy. The entire central nervous system, so spinal cord as well. Yeah, I mean, that makes sense to me, coming from the brain, skin, and nervous tissue. So that is that it? Just nervous tissue and skin? Okay, so then it makes a lot more sense, and maybe I shouldn't be as surprised if I had remembered my dev bio. Interesting. What's the unit of the graphs we're looking at? I don't remember showing... Oh, these graphs. So this is, I believe, an enrichment score. So it's like fancy computational biology stuff, and I don't know how to explain it. If Resta were here, maybe she would know, but I actually don't know. Some mathematical thing that allows you to calculate a relative abundance, just say it that way. Oh man, it's so nice having biologists in chat to have that information. Melanocytes are even closer to neural tissue than other skin cells. They also come from the neural crest. Oh my gosh, okay, so this is really cool. I didn't even consider the shared lineage of skin and nervous tissue. That's awesome. Okay, so I mean, maybe this is starting to make more sense. I'd be curious to see what else this is doing in skin. Okay, so that brings us closer, or actually right up to the paper that we are talking about today, which is Prion as a membrane adapter protein for E3 ligase CCBL1 facilitates, or CBL, I actually don't know which one that is, facilitates the ubiquination of IGF-1R, promoting melanoma metastasis. That's a lot of words, but let's start by defining this first one here. So proprion is not the same as the normal mammalian prion protein. Proprion is actually a variant prion protein that sticks out into the cell cytoplasm. So here on the left, we have, oh gosh, I really need to get a mouse that's a little more stable than the one that I am currently using, because I cannot draw with this thing. On the left here, we have the normal prion protein. This is a membrane anchored protein that is embedded in the membrane, but it does not stick into the cell. And the proprion is we call it an isoform. So this is a naturally occurring, meaning that you have the genes for this. You have the genes to make this, but it's not very common to make this version of the protein, but it is a biological possibility that this protein is made. And this specific isoform is one that sticks out very slightly into the cell cytoplasm. And we're going to see how this sticking out into the cell cytoplasm is going to affect its ability to, let's say, regulate skin cancer. All right. It being a bit wobbly is charming. Thank you. Yeah, maybe. So this is their graphical abstract, and there's a lot here, but we're going to understand what this entire thing means by the end of the day. But essentially, I wanted to show this because their picture is very good. So here we have, on the left here, a normal case where prion levels are low, and this will actually lead to the inhibition of metastasis. And then on the right here, we have a case where the prion protein is in that membrane. You can see it in that membrane embedded in the lipid bilayer. And this is going to act as a protein adapter for this protein complex that is going to lead to autophagy, which will then lead to metastasis. And so if that is opaque to you at this point, it won't be for very long. I will get into it. But for you biology heads out there who are very used to seeing graphical abstracts like this, I wanted to give you a little bit of a preview here. All right. So how the heck did we get here? Prions and melanoma. To me, this is not something that I would have seen coming. And in fact, even though the skin and nervous tissue have that ectoderm shared ancestral origin, I still don't think anybody could have de novo come up with the idea to look at the prion protein in the context of skin cancer. What molecular pathways are most commonly mutated in melanoma? I don't know about most commonly, but autophagy is involved from what I understand. So ULK doesn't get phosphorylated, and that promotes autophagy. I don't actually know how much of this pathway they tested because I had to stop at some point. Okay, dartboard, Dreamless Sleep says. So that's not entirely incorrect, because we start off this story with a bit of data mining, actually. So the authors of this paper took a look at 3 different data sets regarding melanomas. So they have OncoMine here in this, what would you call this, like turquoise? So this is a set of genes that are differentially regulated in melanoma versus normal skin cells, or normal melanocytes. We have this GSE. Pathway, the database that compares the genes that are differentially regulated in metastasis versus the primary tumor. And then a third database that compares early metastasis to no metastasis. And in this data mining, we actually have a set of only 41 genes that are all upregulated in this particular data set. And, you know, I'm not a cancer guy, so when I see this, my eyes completely glaze over, right? Like, I don't know what any of that is, but as a prion enjoyer, I would absolutely freak out if I saw that this protein, the prion protein, in these 3 different data sets, all having to do with metastasis and melanoma. I would be shocked, and I would be chomping at the bit to figure out what the heck the prion protein is doing at this intersection. It's kind of insane to me. Kind of insane. All right. Yeah, I'm pogging. I'm absolutely pogging. Yeah, that's really shocking to me that it came out of a data mine, and very exciting, honestly. So we know that from data mining that the prion protein is potentially involved with skin cancer, but before you invest $1 million into figuring out the role of prions in melanoma, what other questions might you want answered? So this is a good opportunity for, the undergrads in chat, or, anyone who wants to develop a scientific reasoning skill set, a chance to practice those skills. So you have this tantalizing lead. Before you spend a lot of money investigating this lead, what would you want to first double check before you go in to doing this actual research? I went last in your class because I keep falling asleep. No offense to your lesson. No, I totally get it. Again, I have students who fall asleep in my class all the time. And I also, when I was a student, fell asleep in classes. Classes are kind of hard to stay awake in. And I've always felt that I struggle to do so. It's, and there's also the VOD if you want to catch up, right? So that's how I don't take offense to people falling asleep in my classes, because my classes are either recorded in some way, or there are slides or something, and you can always get the information afterwards. We live in current year, of course. I had a very similar reaction when I found out about prion expression in solid tumors. Yeah, it's crazy, right? Because a lot of its reputation comes from its role in neurodegenerative disease, and not a lot in any other field of biology. So really, really surprising finding. Yeah, chat. You have the tantalizing lead. Anyone want to offer one experiment that you might want to conduct before you start researching the role that the prion protein has in melanoma? And I know enough of you are out there where you could give me an interesting question, because I want to embed or imbue a sense of progress in your scientific development, right? So getting an insight into how research should be done, I think, is important, just for kind of enrichment and understanding of why science is done the way it is done. A guy says, do prion knockoff mice have more or less skin cancer? That's an excellent question. That's an excellent question. But I would say that's a little too expensive. There's something potentially cheaper than developing that kind of assay. Maybe not cheaper, but more justifiable than that. More justifiable than that, right? It's certainly something that is an interesting experiment, but before we can confidently do that one, we need to make sure of something else. Maybe whether there's really expression of prion protein in skin. That's good, but a little too far back. Something closer to the cancer question, actually. Fetal bovine serum, cat yogurt. I don't know if you came from the clockwork, not the clockwork, the carbon stream that I was on, but we had played guess the Price is Right for lab reagents, and I had described a lot of our commonly used medias as forbidden Gatorades. Are the cancer cells producing prion protein or just accumulating it? Also an interesting question. These are all mechanistic, which is not necessarily bad, but I'd say that what's more important than any of these mechanistic questions is real life validity. So these are all great answers, and even answers that we will see addressed in the paper, but there's something that should be done much before we get into the mechanism. We should look at, what should we look at, Chad? Something before we take a crack at the mechanism. Is there PRP for real cancer cells? There we go. That is much closer, and I'll actually just take that. And yeah, combination between Clockwork and Leo's most recent answer. We should first look at real people, right? All of those questions and experimental designs regarding mechanism are great experiments that we can definitely do, and a lot of them will be done in this paper. But before we do that, we should check if real people actually have elevated prions in their melanomas, right? So making sure that whatever research we're about to sink hundreds of $1,000s into is actually going to apply to a real human. So make sure that you do that first. This is data coming from 19 patients, 19 low prion protein patients and 19 high prion protein patients, and we can see that this is a survival curve. So this is another one of these pieces of data that reflects the actual loss of life, which is a bummer, but is important when your life's mission, I think, is to tackle one of these horrible diseases. And we can see that in patients with low levels of prion protein, they manage to survive longer, although the p-value is pretty borderline. What are the upward spikes? I think these, I mean, that's a really good question. I actually don't know why they have these little spikes here. I should have looked into it, but I'm not entirely sure. The paper is in the description, and so someone could attempt to figure that out. That would be very helpful. Or if someone knows, that'd also be very helpful. At first I thought they were error bars, but I don't think that makes sense anymore. People resurrecting briefly. I mean, that would be noteworthy for a different biological reason. Okay, so the p-value is 0.050, so it's borderline. The ends aren't that high, but given the magnitude of the difference, I'm willing to believe that this is probably real. So this is another, so this is a cutaneous melanoma, so skin melanoma, and then this is uveal melanoma. And so this one, the p-value is lower, which means that it is at or below the it has achieved the threshold required for statistical significance if we have a p equals 0.05. However, the ends are more than double. More ends is not always the solution, but again, statistics is just number philosophy. This is something that I have become more accepting of, talking to Mylan, who has taught me a lot of about statistics. Oh my gosh, Easter was yesterday. That's true, Morjuice, that's true. Terrible. Yeah, so the difference is there, I think. Like, to me, it ends up being about the same, but that's probably because if you survive for 50 months, you're probably good from there, is what I would interpret this as. But it does appear that with a sample of 40 people, that you do have a more precipitous drop in survival faster if you have more prion protein. So what about the other genes? So we have that list of 41 genes that are upregulated in metastatic melanoma, and we're focusing on prion protein, but you know, there's no really good reason so far why we're doing the prion protein other than that it's cool. So what about those other genes? So apparently prion transcripts were one of the only genes that were significantly upregulated in metastatic melanomas from real patients. Although this data is tricky. So I actually sent this graph to Mylan, and I was like, how... This is a box and whisker plot, and so the lines here, down here, in the center of the bar, that represents the median, and then the actual bounds of these boxes represent quartiles. So this gives you an idea of the distribution of the data, and then the error bars here give you a range, right? So we can see that the metastatic patients, the patients with metastasis, they have a larger range of transcripts. So transcripts are mRNA molecules. So they have more prion mRNA molecules. And according to whatever statistical test they did, this difference is statistically significant, even though the medians it looks identical, and that the quartile ranges also look identical. However, the way that Mylan had explained this to me, and I might get this wrong, so if I get this wrong, don't blame Mylan, blame me. But I think the test that they performed here is something called an independent t-test. So this is a test for testing whether or not the difference of these two data sets is meaningfully above 0. So I'm not enough of a stats person to really fully understand how much stock I should put into this being true. But let's just say that this is not an incredible difference. Don't you love statistics, Fi? I will say that talking to Mylan more, I have been enjoying thinking about statistics more, because it is more philosophy than I gave it credit for. A t-test, but they used a median. This is just a way to display the data, right? So box and whisker was the most appropriate for whatever reason, and then they used the t-test. I don't really like it. I don't really know how good or how quote unquote kosher this operation is. But my first impression, just looking at these data ranges, is that if there is a difference, it's probably not crazy. It's probably not a huge difference if there is a meaningful difference here. Okay. Here's my generous read. Prion mRNA might be hard to extract and measure, right? So this is 300 transcripts per million transcripts. So this is not a super high abundance transcript, potentially, although I'm not like an RNA guy, so I'm not entirely sure, like, relatively how big this number is compared to other genes. But whatever. mRNA, whatever. The important thing is protein, right? And we already saw that patients with high levels of prion protein progress worse. And so when you take biopsies of those tumors, we have the Nevi cells. These are precancerous melanocytes, and then they are different than these stage 4 melanocytes that have progressed to cancer. I'm not a histopathologist. I don't really know what these arrows are pointing to. Whenever I see histopathology, unless it's very obvious, I just smile and nod and go, "Great job, histopathologist, you did it." Yeah, I don't really know what these arrows are pointing to, although I'm sure it's in the figure legend. So the data that they get out of this is represented with this box and whisker plot. This is a lot more convincing, I think. Here we have a p-value of 0, which is kind of funny. I think this is kind of silly, and if I were them, I would just say p less than 0.05 or whatever relevant alpha cutoff we have is, instead of this silly p equals 0, because that's, I don't know, I don't like that. But what we're seeing is that the cells, the biopsies that do not come from highly progressed cancers, this is, I think, a lot more noteworthy, right? So if we have normalized Nevi prion expression at 1.0, the highly developed cancer, stage 3 and stage 4 biopsies are about like 3 times as much prion protein, according to this pathology, histopathology slide. And I think that is a lot more convincing than the RNA stuff. So what the heck is going on, right? So we have this relationship, we have this correlation, I should say, this correlation between the amount of prion protein in a melanoma and highly metastatic cancers. So what the authors then did was they took the profiles of these patients that are expressing high levels of prion protein, high levels of prion mRNA and low levels of prion mRNA, and they're just going to try to align them with other things that are upregulated. And so what they are seeing is that you have high levels of prion mRNA being correlated with melanoma pathways. This is to be expected given the hypothesis of this paper. We also see that the more prion mRNA you have enriched regulation of the active cytoskeleton. So the cytoskeleton is important for cancer, right? Because they need their cytoskeletons to move and invade surrounding tissue. And then insulin signaling. Insulin signaling is also very important for the development of cancer. And then something called macroautophagy, which is a pathway of self-digestion. I will preface this by saying I don't know a lot about autophagy. I just know that it is a stress response that is involved in cancer. So from what I could find, those little marks in the survival plots is often used to indicate patients dropping out from an observation. Oh, the paper itself doesn't really say. Yeah, I found that to be pretty frustrating with this paper, is that there's a lot that goes unsaid in this paper, and it made it kind of hard to read. Okay, so here are the results so far. Data mining shows that the prion protein might be upregulated during metastasis of melanoma. Patients with higher prion protein expression might be worse off, depending on how you interpret those survival curves. Later stage biopsies of melanomas have more prion protein, and prion mRNA is correlated with melanoma actin cytoskeleton regulation, insulin signaling, and autophagy. So this is what I would call the starting base of justification for future study, right? So this is not having to do with anything about mechanism, but if you are trying to justify the spending of hundreds of thousands of dollars into medical research, it's good to at least get a sense that this matters in a real human. Okay, so we have kind of evidence that the prion skin cancer connection is real. What do you want to do next? So I will take a moment to hydrate and ask once again for experiments that you all want to run. You can repeat some of the old ones that you had mentioned in chat already, given the new context of how this paper is staged. Science surrounding autophagy has like erupted since the first isolation of mTOR back in the 90s. Like I wanted to make a video on how tuberculosis maybe messes with mTOR. Oh, Clockwork, can you remind me what mTOR is? I'm having trouble recalling. I know it's cropped up a bunch when I did some of my research into the bird flu, but I actually don't remember what it actually is. Those pathways are so complex in the description now. Yeah, autophagy wasn't really on my radar until very, very recently. Gotta head out, but this has all been very interesting. Thank you for stopping by, Astral. Yeah, so, yeah. Give it a little bit of time to just think about what you want to do next. Master target of rapamycin. I actually only know rapamycin in the context of like genetic engineering. I'm not health pilled enough to know what physiological role of. I guess that's probably how they discovered mTOR, but I'm actually not sure what mTOR's physiological function is without having to look it up. Y'all, there are a lot of things in biology. So if you're like, I can't believe it doesn't know what mTOR is, there are a lot of things. A lot of things in biology. Balances growth / autophagy signals and is a big metabolic regulator. Oh, interesting. Yeah. I'll because I'm more involved in like the pathogen literature, I don't really cross over into the cancer stuff too often. I do wonder if melanoma cancer cells survive oxidative stress better. Oh, I wish I could tell you, because cancer cells have a very interesting relationship with oxygen. I believe we learned in the 12-hour glioblastoma stream that a hypoxic environment, a local tumor environment, a local microenvironment that is hypoxic is beneficial for cancer cells. So certainly oxygen is quite involved in their story. There's a great explanation of mTOR in a recent Chubby Emu video. The problem with making content is I often don't want to watch other biology, other like health / biology VTubers, not VTubers, content creators. I would actually prefer to read about mTOR, not like out of a sense of competition, but like watching someone else's biology video and like having a similar idea and then having it scooped from you is like very frustrating. Warburg effect. Cancer cells can do anaerobic metabolism, so weird relationship with oxygen. Yeah, that's how I would describe it as well. Like, I'm not confident enough to say if they survive oxidative stress better, but I will say, like Guy said, their relationship with oxygen is complex. All right. I think the collection versus creation of prion protein is interesting. If the cancer just collects it and IDK uses it as a shield against something, and it might be useful for some treatment. Oh, Leo, keep that in mind. Especially that last part. You're inadvertently spoiling something that we will come across at the very end of the paper. So this one is slated to be a long one, so you might not make it to the end, but the idea of the prion protein being a treatment, it's interesting. It is interesting. All right, so I'll just go with some of the experiments that chat had recommended earlier in the stream, and we'll just continue along right here. So the first thing that the authors want to do is play with their prion construct in a cell line of melanoma. So here we have two different cell lines, A375MA2 and WM266.4. I don't know what the difference between these melanoma cell lines are. For most of the rest of this paper, I'm only going to be focusing on one type of cell line, just because I don't think we need that level of nuance. And so the way that we tell whether or not we have a successful knockout in a melanoma cell line is, drumroll please, the Western blot. The Western blot, yes indeed. Should have guessed it was a Western blot. It always is. It always is. That is so true. This is why Western blot is an S tier method, even in the current year, 2025. I think this was in 2024, this paper, but like, even though this paper was invented so many decades ago, it is still extremely useful, and we will see how incredibly helpful this technique is, even though there are fancier techniques out there. So what are we looking at? So this is my Western blot 101. If you have attended a couple of these streams, you don't really need this recap, but I will do it anyway, just in case people are new here. So first thing that happens in a Western blot is that proteins are separated by size with an electric current. Proteins are then moved to a membrane. So they are moved to a membrane in a kind of ladder orientation, where you have the larger proteins higher up in the ladder and the smaller proteins in the lower end of the ladder. You slap on some antibodies that are specific for a target protein attached to some visualization group, like an enzyme that produces color or an antibody that produces color. And then the darker the band is, the more protein. This is all normalized to what is called a housekeeper gene that is not predicted to change in the experimental conditions. So the Western blot is what is known as a semi-quantitative analysis method. This means that I can't tell you that there is X amount of prion protein. Like, I can't say this is 26.2 nanograms of prion protein, but what I can say is that this band over here, in this condition where you have the regular prion producing melanoma, that there is more prion protein than this cell line that lacks the prion protein, right? So that's the difference between this column and this column. This column has the prion protein genetically expressed, and this column is the same cell line, but you removed the expression of the prion gene. And so we can see here is that we have a very, very clean knockout, which is wonderful. So we know that this is a cell line that truly does not make the prion protein. I might have to do 6 blots simultaneously tomorrow. Pray for me. That's insane. That's insane. Gonna do my first Western blot in a few days. Good luck. Very happy for the recap. That's so exciting. I love the Western blot. Housekeeping gene in these results is actin. Yes, actin. And that's an interesting... I wonder, Clockwork, if you pointed that out for any particular reason. And I'll give you a chance to... I don't know. Because I am going to talk about actin, but I don't want to... I want to... If you have something interesting to say about actin, Clockwork, then that would be good to know here. Skin. So I'm not entirely sure what you mean by that, but I will go on. There is an interesting idea of... An interesting thought that I had that might not be that important. So, we have any comments on how this looks? I don't know how many of you are actual biologists. I told you that the darker the band, the more protein you have. That's certainly true, but this one's for anyone who has run a Western blot. I don't know if any of you are in the comments. But does this band look funny to you? Even if you're not a biologist, compare the shape of this blot to the actin blot. Is there anything weird about how these shapes are in comparison to the very clean actin band? Why is it so long? It's smeared AF? Yeah, why is it a smear? Yeah, exactly. That's kind of interesting, right? Hey, Yan Yan, welcome. What I miss? We are talking about the role of prions in skin cancer. We haven't actually gotten that far into the real paper, but we did begin with a conversation about why the prion protein continues to be evolutionary conserved. Yeah, so I will go in order of just the slides just to keep it convenient. Yeah, this band is like really smeared. It's really bizarre. I wonder why that is. So my hypothesis is that we do know that the prion protein has isoforms, and we do know that the prion protein gets cleaved, right? So after the prion protein is made, it can get modified in all sorts of different ways. Things, parts of the prion protein can be chopped off, parts of the protein can have additional chemical groups added to it, which will actually affect the prion protein and give us a variety of different prion species. I don't know that that's the case here, but you can actually see, if you look very closely, a couple of distinct bands that might correspond to specific forms of the prion protein, which I think is interesting. And then the second question I have here is actin actually a good housekeeping gene here? And so yeah, a couple of you have got it in the chat. Didn't we previously mention that the protein has effects on the cytoskeleton? We know that the prion mRNA level is correlated with actin regulation. That's how I will say. So not exactly the protein, but the gene, right? Cytoskeletal modifications are the function of metastasis. Yeah, so there's some confounding variable here. Although by eye it does look like these actin housekeeping levels are about the same, so it's not, you know, it's not egregious that they did this, I think, but it is an unusual decision. I wonder why they didn't use the more common housekeeping gene GAPDH. Maybe GAPDH is also involved in cancer. It's just actin seems like a very bizarre choice for a cancer housekeeping gene, because we know that actin is changed in cancer, put it that way. Yeah, isn't that interesting? Just from one blot, we have two interesting observations. Multiple bands and actin as a housekeeping gene is kind of interesting. Okay, very, very cool. So, knocking out the prion protein decreases wound closure and decreases cell invasion. If you watched my other cancer streams, you would kind of recognize these as metrics of migration and metastasis, right? So wound closure, so you can plate your cells, you can drag a blade through them and see how quickly the cells patch up the damage in their network. And we can actually see that in these cell lines that if you don't have the prion protein, you are worse at repairing those repairing that damage. Similarly with invasion, I'm not going to go over how this was done because I didn't have time to really figure it out, but this is very dramatic, right? Losing the prion protein causes a enormous precipitous decrease in the ability of these melanocytes to invade neighboring cells. So that, I think, is really, really strong evidence for the prion protein playing a mechanistic role in metastasis, at least in culture. GAPDH is pretty close in size to the prion protein, I think. Oh, yeah, that's probably why. Yeah, so for the housekeeping gene, you need a protein that does not overlap in size to the prion protein. So if GAPDH and the prion protein are similarly sized, yeah, that's not going to work out. I'm surprised. I thought GAPDH was pretty big, but I guess it's... Wait. Yeah. If they're the same size, you can't actually have that happen. So that's why they didn't choose GAPDH, but I'm sure there's another housekeeping gene they could have found that wasn't implicated in cancer. Yeah, so just to recap, removing the prion protein from skin cancer cells in culture causes them to be defective in this way. So, melanoma cells lacking prions don't metastasize as often, and here is my content warning, because we are going to look with our very eyes an actual culture of melanoma, right? So we're going to see something fleshy. So, you know, if that makes you squeamish, avert your eyes in 3, 2, 1, changing slides. So the top here, we have a chunk of cells. Actually, this might not be a cell line. This might be a lung. That they like grafted melanoma to. I don't actually quite remember because I had to blitz through this paper. I had so much to talk about. But the idea here is we have some sort of cell system up top here. Flesh in my VTuber stream, more likely than you think. And then bottom down here, we have the cell lines that are lacking prion protein. And in the green circles, we have metastasis. So again, I'm not a pathologist, so I just look at this and go sure, but you can see these like white nodules lump up from what is otherwise smooth tissue. So I do find that to be interesting. Yeah, they might be lung. I don't know if this is 3D cell culture. I have 110 slides to get through, but do know that I'm not entirely sure what this system is. If you measure the tumors, you can see that in the I'll call it the system. These look like weird bloody oats. They actually do. It's disgusting. Why they grafted into lungs? Maybe because they needed a system that actually functioned within a living organism. You know what? Maybe I'll just look it up real quick. Chat not expecting flesh in a biology stream is the more surprising part. That's true. Although I'm like, I'm not the touch grass kind of biologist, so I don't typically show flesh. My streamer showing flesh. So let's take a look at this caption. No, it is cells. This is cells. Yeah, that's so weird. Like I've never grown cancer cells. I didn't even know that they can grow in 3D like this. Yeah, the caption says effects of knockout PRNP on melanoma cell metastasis in vivo. Although, hold on. Well, now it's bothering me. Give me a second, because we have a different figure down here. Yeah, I think it might just be cell lines. I don't think that this is lungs or something, because they do lungs later on, actually. For sure they do lungs later on. Yeah. I'm expecting graphs and weird abbreviations. Yeah, that's the kind of biologist I am. Pathology has no life is best spent in hospital basement. The only light I need comes from my microscope. I would, if medical school didn't require such a big, like, memory bank, I think I would have enjoyed being a pathologist. I think that would have been a fun alternate life for me. But yeah, so I think this is pretty striking, right? If you don't have the prion and you are an isolated group of melanoma cells, you are not doing metastasis quite as often. And this actually does look quite good, right? P-value is low, difference is quite big. It's quite striking. So I'm very careful here to point out that these are melanoma cell lines. And Chad, do you have an idea of why I might be this careful with my language? Like, why am I being so careful to repeatedly call them melanoma cell lines? What might I want to be avoid? What might I be avoid? Ugh, what? I can't speak. What might I be avoiding by calling them melanoma cell lines? Yeah, Clockwork. I think like microscopes are great because melanoma cells evolve very rapidly. Not quite. I mean, kind of, but not really. It's more to do with the last cancer stream I did with the paper that shook the world, the very call-outy, angry paper about the genetic model of cancer. And yeah, and Rand's got it. Cell lines often aren't representative of how cancer cells actually behave inside an organism. Right, exactly. So this works in a dish, which is not bad, right? They're not bad. This is not an experiment that I see and go, oh, this is a bad experiment. It's just by calling them cell lines and being careful about calling them cell lines, I am reminding myself that I am not talking about real cancer. I'm talking about kind of a sandbox cancer, right? Something that you can play with in a sandbox. So it's a way of me hedging going too deep into buying what the authors want to sell me. In vitro data useful, but not as complete as in vivo. Exactly. I actually don't think that's a hilarious oversimplification at all. I think that's exactly right, right? Like in vitro is useful, but it cannot form your entire understanding of a problem. Kind of how pudding forms a skin only if exposed to air, while being only exposed to other pudding won't create any skin. I'm gonna be honest, that caught me off guard. I actually just imagined like an actual human skin layer onto pudding, and that was not an image I was ready to generate in my head. Disgusting. Okay, but yeah, so as the comments have pointed out, this is not to say that experiment bad, but experiment not complete. So if there's one thing I've learned about cancer is that the tumor microenvironment and the surrounding tissue is very important. So we're going to keep that in mind for the rest of the paper, just so that we do not believe too much, right? That we don't buy something that is not on sale, we'll say. So here are the results so far. Melanoma biopsies from humans have more prion protein in late stages compared to early stages. Melanoma cell lines, important distinction, without prion proteins are bad at cell wound closure and metastasis. What's the next step here, right? So these are the simplified versions of the previous data, and I want to know, where do you think we will head from here? So I would say that at this point, right, we are pretty sure that prions are doing something, that they are potentially affecting skin cancer in a negative way. What is our next step experiment? What do we want to know? Also, welcome to the stream, Toxo. I never fail to be amused by the 5-ohm emote. It's so good. PRP knockout says Clockwork. What do you want to know about the PRP knockout? We already have pretty good justification that this matters. And so what might we want to know about a PRP knockout? In vivo verification, that's actually a very good step that they could have done. They don't really do that until very late into the paper, but they will do so. That's not their next step. That's kind of how they wrap up the rest of the paper. So Clockwork and Ran, you are right that they will be validating, but they're actually going to validate as a way to tie up this entire story in a neat bow. So that is coming, but it's not the next step. What else? What else? What might we want to know? We know that it's important. We know that it happens in real people. And if we are biologically minded researchers who are fighting for funding, especially in the current climate, what might we want to actually try to find out besides verifying these results in vivo? Don't get me wrong, verification is very, very important, but it's, in this particular case, not really the moneymaker. What is the moneymaker here? What is the moneymaker for most biology, molecular biology research? Yeah, kind of an interesting question, right? What is the next step? Well, if you are doing molecular biology research and you want to be flush with cash, although again, in the current climate, it's a little difficult, but traditionally, you want to know how this is happening, right? So how is the prion protein connected? What is it doing in the melanoma? Essentially, that's it. How is this happening? And so a prion knockout is going to be very, very important. And so inspecting how the prion protein is functioning in these cancer cell lines is going to allow us, as Clockwork is correctly saying, something that you can sell, right? Making a treatment, making a drug, right? That's the moneymaker here. We already have a clinical correlation ran from the clinical data showing that patients with higher levels of prion protein in their melanoma biopsies have worse clinical outcome. That's pretty good justification for trying to attack the prion protein in skin cancer. But in order to make the big bucks, you got to make a treatment. And to make a treatment, you first have to know what the heck this protein is doing. And so we are going to be talking about the mechanism, right? So you can kind of see that there's an arc of storytelling happening here. First, you have to justify the problem. Then you can figure out what the heck is happening. And then you want to remind people at the end of the paper that this actually does matter in vivo. So these are all important parts of the story that these authors are trying to tell. So we are trying to find out how the prion protein is causing cancer. And one approach that we can take is co-IP. And I'll get into that in a second because I just saw a great question from Gabrielle. Does the melanoma cause the prions or do the prions cause the melanoma? That is an excellent question. We actually do know that when we remove the prion protein from the cancerous cell lines that we grow in a dish, that they do not metastasize as often. So that is an indication that it is the prion that leads to the melanoma and not the other way around. Although biology is a spectrum, you can argue that maybe there's some shared condition that causes there to be some sort of correlation, so maybe it's not entirely one-to-one, but in general, with that clean knockout, we do see that in a dish, you stop skin cancer from metastasizing so hard when you remove that prion protein. Yeah, so the approach that the authors take here when asking the question, what the heck is the prion protein doing in skin cancer, is through a protein lens. And if you love Western blots, we are going to look at so many of them. But in particular, we are going to be combining the Western blot with a technology called CoIP. You can do !CoIP if you want a recap for what that is. You can try out the new glossary command if you want a recap on any of these definitions. So the way that a CoIP works is you have a bead, or you have a set of beads, a lot of beads. Oh my gosh, it's so hard to draw with my mouse. You have beads, tiny, tiny beads that are attached to antibodies. And then these antibodies, you are going to flow over cell lysate. So you're going to take your skin cancer and you're going to mash them up in a blender and pour the juice over these antibodies. And the antibodies are going to bind to their antibody target and whatever the target protein is also binding. So you can see we have three different parts, right? Bead here, we have an antibody, we have a target protein, and we have everything that the target protein binds. Those are the parts of a CoIP. So when you capture the binding partners of this blue protein here, you can then shove them into what is known as a mass spectrometer and unveil the identity of all of their binding partners here. And that's going to give us a lot of good information as to what the prion protein is potentially doing. So, any questions on the CoIP? It's actually pretty important to be on the same page here because we are going to be spending a lot of time talking about the CoIP. So, just going to start a poll real quick while I do another little hydration. Oh, talking a lot. I missed talking to you all. I really did miss talking to you all. I'm so excited to get to do that by talking about one of my favorite subjects ever, which are prion proteins. But yeah, let me know in the poll if the idea of the CoIP is good to you. Oh, it's good hydration. All right. All right, it looks like we're mostly okay to continue, but I will explain it just one more time. So, in order to identify all of the prion protein's binding partners, we are going to be using a technology called co-immunoprecipitation, or rather a technique. By finding what the prion protein binds to, we can gain mechanistic insight into what the prion protein is doing in skin cancer. So if we find out that the prion protein binds a major oncogene, oncoprotein or something, then we can start making conclusions on what the prion protein is doing in skin cancer. So that's the purpose of this experiment. The way it works is that we have beads that are attached to antibodies, attached to the prion protein. So this is a bead right here. And then we have this, which is an antibody against the prion protein, so it binds only to the prion protein. We're going to throw on just the guts of the skin cancer cell, so all the things that are in the cytoplasm. And that's going to bind prion protein. And the prion protein is going to come attached to whatever it happens to bind to. And by finding out what it binds to, we can figure out what it's doing. Okay. Alrighty. Cool. So let us continue. Okay, so mass spectrometry. So we are doing the CoIP, which allows you to identify all of the prion protein's binding partners, and then we are going to shoot it into this very complicated machine called a mass spectrometer. I am going to do a 101 version of this that is very, very simplified, but I think it's cool enough to try to explain here, right? So a mass spectrometer that you use for protein identification works like this. You take your cells and tissue, and you blenderize them, and you isolate just the proteins. And then you digest the proteins into peptides. The peptide mixture gets shot into this tube, which ionizes, it gets shot into this tube which separates them by size. So it filters them with liquid chromatography. And then this gets sprayed and ionized into ionic peptides, so ionized peptides. And then these peptides crash into a detector and give you a measurement. So to simplify this, what I'm going to say is that The principle by which the mass spectrometer works is that every peptide is made of a series of amino acids, right? Like glycine, serine, threonine, arginine, right? That's a peptide. And so the way that a mass spectrometer works is that it charges up that peptide and gives all of the available charge that peptide can hold, and then that crashes into a detector that allows you to measure the ratio of the mass of the peptide to its charge. It's a little complex. This is more for my Biology 101 undergraduates, but I hope that's clear, that we are identifying protein mixtures, breaking them up into peptides. Because every peptide in existence has a unique mass to charge ratio, you can actually layer on those peptides back into proteins and figure out what proteins were in a sample. What did I miss? You missed us briefly talking about prion protein conservation across mammals, which is pretty interesting. Definitely recommend watching the VOD, because I think that understanding that the prion protein is more conserved than its doppelganger protein in mammals is actually pretty interesting to think about. And then we talked about the fact that prion proteins are upregulated in melanomas from actual human patients. And now we're trying to ask the question of what the heck the prion protein is actually doing. And so to do that, we are going to be doing a co-IP, trying to figure out what the prion protein binds to. And there are two parts of this, the co-IP and the mass spectrometer, essentially stitching together the identity of every protein in a sample based on the charge mass ratio of its peptides. And if you didn't understand that, that's actually fine. It's not that important. I just think that if you are like physics or chemistry minded, that this might be of interest to you. Hello, welcome, Wild Max. Okay, so what proteins emerge from this complicated... Oh, by the way, this is something that I wanted to say. If you are a biology student or a chemistry student or a physics student and you want a pretty stable job, join a lab that has one of these things and then figure out how it works. Get trained on this thing, become a mass spec technician, and you will make so much money and always have a job, because these mass spectrometers break so often. And I had the displeasure of working with one. And for most of my rotation in the mass spec lab, the thing was broken. So someone has to pay to get that fixed. And if you are the person that fixes that mass spectrometer, you can take home a lot of money. So when people say there's no money in biology, there is. It just depends on where you're looking. Get trained on this thing. It's good. Yeah. Alrighty. So what is the prion protein doing in skin cancer? Well, we can address that question by doing the co-IP followed by mass spectrometry. And what the authors found was that there was this one protein that was almost always coming off with the prion protein. So if we kind of imagine this as like a fishing rod, this fishing rod is always fishing out this protein, which is CCBL. So this is a protein that constantly is being fished out by using the prion as a fishing line. So this is a consistent binding partner to the prion protein. So what is this protein? CCBL1 is an E3 ubiquitin ligase. I've actually talked about E3 ubiquitin ligases before, actually. I actually don't remember in what context, but I've definitely talked about E3 ubiquitin ligases. I think in the context of Ebola. These are enzymes that attach a ubiquitin tag to proteins to mark them for destruction. So ubiquitin is a tiny protein tag that ubiquitin ligases attach to proteins, and that gives the protein the correct shape to enter what is called the proteasome, which is a protein complex that is kind of like a garbage disposal. It's like a horrible sarlacc pit of degradative chemical reactions that chew up proteins. So you can think of CCBL as a protein destroyer. So the prion protein is consistently binding a protein destroyer. And so how was this confirmed. So that was by mass spec, right? That's fancy computer technology, fancy billion dollar machine, but you still got to confirm it with a Western blot. So co-IP was performed. And so up here you have the antibodies. So up here you have the anti-prion antibody. And here you have the IgG1, which is a antibody. This is an antibody that's not expected to bind CBL. So it is our control. And so we have our IP product down here. And up here, we see the, gosh, again, it's so hard to draw with a mouse. Up here, we have the blot for the protein destroyer. And down here, we have the blot for the prion protein. So what we can see here is that when we use the prion antibody, 8B4, of course, you fish out the prion protein. That's why there's a ton of this prion protein that comes up, right? There's a ton of this. Of course, it's the antibody for the prion protein. It's going to fish out the prion protein. What you can also see is that it also fishes out CBL, and that's confirming what we learned from the mass spec. Does prion binding inhibit CBL? We are going to talk about what that does in just a bit. You are getting a little ahead of us right now. Smear strikes again. Yeah, I mean, I think it's because there are so many forms of the prion protein. If the binding inhibits the garbage collector, garbage starts piling up and the cell drowns in garbage. That would be true if it is inhibiting the garbage collector. But actually, what we are going to find is that it is not inhibiting the garbage collector, but activating it. So I guess I'll spoil that just in case you have to go. But the prion protein is not inhibiting this enzyme. We will find out that it is actually helping it in some ways, which I think is pretty interesting. It is helping the garbage collection. So this is an important control. The prion protein does not fish out, CBL is not fished out when the prion proteins have been knocked out, right? This is just good, clean controls, right? Like before we had here, the IgG1 antibody control, where you use an antibody that does not bind prion, and when you use a fishing rod that is not very specific, you barely get any CBL1. There's a little bit, but not that much. Here's another control. Let's remove the prion protein from the cell line and repeat the fishing rod experiment, right? So we're using the prion fishing rod, and when you use the prion fishing rod, you get prion protein, right? Prion protein. You also get CBL. Duh. That's what we just saw. That's obvious. But when you genetically knock out the prion protein, you no longer fish out the prion protein. You no longer fish out the protein destroyer. That's just a nice control. And clean, right? This is very convincing that this is not some non-specific thing that's happening. This is specifically using our prion fishing rod to fish out the prion, which is going to be attached to this protein destroyer. Slightly irrelevant question, but how do prions transmit? Because apparently they are not alive, but are transmissible. From what we know, they can be ingested and they bioaccumulate in the brain. So they survive all sorts of nasty weather conditions. And I don't know the entire pathway by which they accumulate in the brain, but they can accumulate in the brain. Okay, so prions bind this protein destroyer. What's the next question? What question are you thinking about now? Because this is not a complete story. This is us understanding that the prion protein is potentially anchoring a protein destroyer or putting it in proximity to the membrane. So what's the next natural follow-up question? What do we want to know next? Give myself a break. Wow, it's been a while since I did this much talking, and it's pretty hard on my voice. I'm surprised because taking 1 week off isn't really that much time, but I find myself back to not being able to yap for that long. What does this binding do to the cell would be next IMHO. That is one possible way to go down. That's a little harder to do than what they're about to do next. That's something that they will eventually do, but it's not the next step, Leo. But they're going to get to that. They are going to get to that. So it's binding a protein destroyer. What is the next question? This is a question that you don't have to think too hard. Don't have to think too hard about this follow-up question. You might be overthinking it if you are struggling to come up with the next experiment. How do they unbind, if that is possible? That is assuming that we care about them unbinding. Did you say they also attach to the membrane? So the prion protein is attached to the membrane, and what we know is that this is also binding to this protein destroyer. So this is anchoring the protein destroyer to the membrane. Does the misfolding of the prion disable the prion destroyer? So we're actually not going to be talking about misfolding of the prion protein at all. This is all a feature of the normal healthy prion protein, which is very surprising. So aggregation actually doesn't matter. We're talking about the base prion protein having a cancer function, which is even more surprising. Yeah. So this prion protein anchors a protein destroyer to the membrane. What do we want to know next? You're going to be I think you're going to be kind of mad if I give you the answer, I think. So I really want someone in chat to get it, so I don't take it away from you. So, because I think it's going to be like a facepalm kind of moment if I give you the answer. Ah, yes, Wildmax got it. Wildmax got it. Wildmax got it. What does the prion destroyer do after being anchored? That's it. That's it. Thank you so much, Wildmax. What's it doing? Again, it's not a very complicated follow-up question, but what's the protein destroyer destroying, right? What's this thing doing? Why? Like, so the prion protein is anchoring this protein destroyer to the membrane. What's it actually doing, right? What the proteasome doing? Exactly, more juice. Exactly. Like, so we have these hints. By itself, knowing that the E3 ubiquitin ligase CBL1 is binding the prion protein, that's interesting, but we don't really have a clear connection to cancer. We have to first understand what the protein destroyer is actually destroying. Great. So we know from previous research what CBL1 CBL likes to bind to. Interestingly, in this system, only one candidate is binding, which is this receptor, IGF-1R. So this is another one of these co-IP Western blots. So let's look at our fishing rods. So this is a fishing rod for the protein destroyer. And we can see that when we use this fishing rod, that we in fact fish out what we're binding to, right? The CBL antibody binds the CBL. That's what this is saying. Duh. So our fishing rod is working correctly. But we also see that this fishing rod also fishes out this receptor IGF-1R. And if you are a biologist, these letters should activate some neural circuits in your brain. IGF should come to mind when you think about certain types of cancers. Does darker line mean more binding? It means there's more protein there. So the setup is a bit complicated. I might not go back into it entirely, but in this case, all you need to know is that the darker the band is, the more protein is present. And the way the experiment is set up is that we are using these antibodies, so this antibody and this antibody, as fishing rods. So when you fish out, so this is a fishing rod that is specific for the protein destroyer, CBL. And when you use that fishing rod, you also fish out the IGF-1 receptor. Yeah, dreamless sleep says growth hormone. Exactly, the IGF receptors. This is an insulin-like growth factor receptor, and that is a receptor that is involved with cell growth, which is, of course, something that is involved in cancer. My research was on VEGF, the vascular endothelial growth factor, so that's the one I know better. Yeah, whenever you see GF, that feel when you see GF, you should think growth factor, you should think growth and cancer. So IGF-1R is an insulin-like growth factor receptor. This is a receptor tyrosine kinase. This matters if you're a biologist, but if you're not, don't worry about it. This serves as the start of intracellular signaling. So this is a receptor that is at the membrane surface, and in response to binding of growth factor, they activate cellular pathways that respond to growth, development, and cell motility. So this one signaling receptor is capable of controlling quite a lot here. This is often upregulated in cancers. or I should say dysregulated, as we're about to see. It's not always upregulated in cancer, it's often upregulated in cancer. Some cancers downregulate this, so it's better to say dysregulated in cancer. Oncologists will know GF, true. Can confirm a GF induces growth in me. Nice. Very good, very good. All right, so the protein destroyer CCBL binds to this insulin-like growth factor receptor. Is prion protein mediating this interaction? So we have a tripartite system with three components, right? And so we know that the prion protein does bind this, but do they all bind together? That's the question here. Prion protein does that's not at all more juice. I think chat is often funnier than I am. That's so good. Prion protein does appear to bind IGF-1R, suggesting that perhaps we got a trimer on our hands, or three proteins binding to each other, right? So here we have, again, another of these Western blots. We have the fishing rod for the prion protein. Of course, you fish out the prion protein, and you also fish out the growth factor receptor here, right? And then the control antibody doesn't fish it out at all. Well, just joined during my lunch. WTF is happening. We are talking about trimeric interactions between the prion protein and these other things. So healthy prion protein, one of its forms can stick out into the cytoplasm very slightly and mediate the interaction between this growth factor receptor, which is going to be implicated in cancer, and this protein destroyer. So the way that we're going to see this happen is that the prion protein is actually going to affect cancer by destroying this growth factor receptor, and that's really cool. IGFR1 is also connected to mTOR, which Clockwork was talking about previously. I will have to look more into it in the future, because I think I'm missing a huge part of the story if I don't have that context. So, one final co-IP shows that we almost certainly have a trimer, right? So, I had said before that, we had the co-IPs that show this interaction exists, right? That interaction exists. We also see this interaction existing. One more shows that when you fish using the growth factor antibody, you can actually fish out the protein destroyer when you have the prion protein, but when you don't have the protein, you don't end up fishing that out. So this has all three parts: a fishing rod that binds the growth factor receptor, the lack and presence of prion protein, and the third component, the protein destroyer. If you were not able to understand, that's okay. These take a while to learn how to read. I can also go through them again if you want an explanation, because we're about to head into an optional part of the stream where we talk about how we can actually use Western blots to pinpoint exactly where, with pinpoint accuracy, where on that protein this interaction is happening. So I'm going to throw up a poll here, see if we want to talk about more about Western blots. Do we want another explanation for the co-IP Western blots? Yeah, it'll give me, again, another bit of time just to rehydrate here. Yeah, because there's that optional section that's coming up that is going to be very Western blot heavy that we can just skip over if we're tired of looking at these things, which is completely fair and fine. Damn, there were required parts of the stream. I missed a bunch at the beginning. Am I going to fail? No, Toxo. Of course, you can never fail. You can never fail. As long as you are showing up, everything is good. But there will be a quiz at the end to see who I love the most. All right. Let's see. Okay, another explanation for the co-IP Western blots. No problem. We will do so. Okay, so let's go back a couple slides. So this is the setup for the co-IP. Let me go ahead and get that pencil. So the co-IP consists of several parts. You have a bead attached to a fishing rod antibody. This antibody is going to be specific for a particular target. You can think of this as like a lure, right? If the antibody is a fishing rod, the protein that it is designed against is like a lure, and that lure captures binding partners. All right, so there's 3 systems, or a couple different aspects going on here. So the co-IP experiments are all about fishing binding partners. We are trying to figure out inside the melanoma what proteins are binding what. And when we crack open the cell, these protein interactions are still happening. So when we use antibodies as fishing rods to pull out the lure and pulling out whatever fish we catch, that's how we know proteins are binding. So when we look at something like this is a Western blot that is done after co-immunoprecipitation. And we know from a Western blot that the darker the band is, the more protein exists. So in this column, we'll just ignore the control column right now. So, ignore that. Let's just look at this column. This is the anti-Prion antibody. It's called 8B4. And when we use 8B4 as a fishing rod, we capture a ton of the lure, which is the Prion protein, right? This is the anti-Prion antibody. We get a lot of the lure out. And this lure is also going to be capturing this fish, which is the CCBL protein. Does that make sense, chat? Let me maybe... let me... Yeah, I'm not good enough at writing with my mouse to actually label this. But I'll go ahead and I'll put up another poll just to see if that made a little more sense. So do we understand the co-IP here? And if you have specific questions about the co-IP to enhance your understanding, I'm very happy to take the time to do so. So the 8B4 is an antibody made to reduce prions? No, it's an antibody that's used to bind prions, right? So there are medicines that involve using antibodies to block or stop a protein from doing some bad function, but this is an experimental, by experimental I mean used for research antibody that is used to bind prions, so identify whether or not a prion is there. What do PRP and CCBL1 mean? So CCBL, you can actually do exclamation point glossary to get a definition of that, but the PRP refers to the prion protein, CCBL refers to this protein that is a protein destroyer. So CCBL is a protein that attaches a ubiquitin tag to a protein and marks it for destruction. All right. So, all right, we got a pretty good ratio of yeses to nos here. So I will try to incorporate more understanding of the pull down co-IP as we continue looking at future blots. Okay, mutate certain targets of the known protein binders and see if they are still present in blots when you just purify those mutants. That's an excellent suggestion of Clockwork. It's actually an experiment that I wanted them to do, but they don't do that. They don't do that exactly. I want them to, though. So we are in agreement that that's what I would have wanted to see. But they are taking a very, very, very slightly different approach here. Yeah, so the question that we want answered is that this is how we know that this is an actual tripartite interaction. When the prion is genetically present, when the cell is making prions, you can use the prion antibody to fish out both of these proteins, right? Using the antibody against this allows you to fish out both of these proteins, which is great. And then when you mutate the cancer line to not have the prion protein, you no longer get these two binding to each other, right? You can't fish out these two anymore without the prion protein presence. So that's pretty good proof that they're binding in I almost said an unsay-so word. They're binding in 3 ways. We'll put it that way. Here's some interesting fluorescence data showing their colocalization. So unfortunately, we don't have a 3-color colocalization. This is just kind of weird and interesting to me, where in this, I hate it when people do like red and green fluorescence. It's always so annoying because of color blindness. But we can see here that at the membrane, this is a confocal image, so this is a slice through the cells. So what you're looking at is a top-down view of cells that is essentially being captured at one optical slice. So you can see the nucleus because you can imagine exactly one slice of the cell being shown to you. And we can see that at the membrane, at the outside here, you see a lot of yellow dots that represent the overlap of green and red. So you can see that at the membrane, there is a lot of co-localization of the prion protein and this insulin growth factor. For the protein destroyer, however, that co-localization is a lot more polarized for some reason. Like, it's mostly like here and here on polar opposite sides of the cell, and I honestly don't know why. I just think that's pretty interesting. I don't really have any comments besides I don't understand. But yeah. So here we have the optional slides, and here I will take a poll, another poll, to determine whether or not we want to do these optional slides. And here I'll be a lot more slow and walk through them because I think these are like pretty meaty and pretty nuanced, and I think they're really, really interesting and really showcases how elegant molecular biology can be. So yeah, optional slides, yes or no? How much of a challenge does chat want? Because these are a little harder than your average Western blot, right? Western blot, you just go, I'm looking at a protein, I'm looking at a blot. The darker it is, the more protein there is, and then you move on. Co-IPs, you also have to pay attention to your fishing rod or the antibody and lure that you are using to fish out protein binding interaction. I like Elden Ring, so I'm made for challenge. That's true, that's true. Elden Ring enjoyer. This is probably no surprise for anyone who likes that game. Again, 14, unanimous votes. 15 we got now. Okay, so again, this is so great. I actually cited all of you in a grant that I had written for science communication, being like, my audience wants to learn this complicated stuff that you could not sell as pop sci, and I think that's incredible. You're all pretty amazing. I'm amazed every time, especially because these are a bit tougher. So we understand the basic gist of the co-IP, that you're using a fishing rod antibody to try to find out what proteins are binding. So this is kind of what Clockwork was referring to. This is a series of different prion constructs that are going to be tested for binding to these proteins. So here on this end, we have the outward facing end of the, so sorry, let me back up. What you're seeing is a very simplified cartoon of the prion construct, right? So the blocks, these are just different regions of the prion protein, and then the dashed lines represent places where that block of the prion protein has been eliminated or deleted. So this end is outside facing, so this is outside of the cell, and then here we have this cytoplasm facing. So you can kind of think of the prion protein as this cartoon. So there are a lot of different constructs. So while this might not be an alanine screen as Clockwork was referring to, this is pretty laborious. Each of these mutations had to be made and put into a cancer cell line, and this is a lot of work. So they had to be very selective with what hypotheses they wanted to test. But essentially, they were breaking and removing different pieces of the prion protein to check exactly where this interaction was happening. And this is going to be really important. It might not be obvious now, but later on, this is going to allow us to make medicine, and I think that is why we need to talk about this now. Looks like I have an error message. Am I still live, y'all? Please let me know if we're still good here. I think I'm good, right? Everything fine? I think I'm good. Okay. Yeah, that was weird. Okay, OBS just freaked out. Cool. Right. Thank you so much. Yeah, so there are a couple of different versions of the prion protein that these researchers made from scratch, right? So you have flag PRP. Let's see if I labeled these. I labeled these, right? Yeah. Okay. So flag PRP, this is the normal prion protein. And we added a flag tag to it. That's this red bar over here. right? That red bar, that's a flag tag. This is appending a bit of protein at the end. That is a very convenient way to purify protein because there's a very good antibody against the flag region. So this is not the prion protein. This is adding something extra on top just to make purification and stuff like that a lot more convenient. And here you can see a region of the prion protein called the GPIPSS, which I'm not going to get too deep into, but this is a region of the prion protein that is important for signaling that sticks out into the cytoplasm. How do you add that tag? Genetically would be the most straightforward answer that I could give. I don't really know exactly what method they use to add the flag tag, but you can do things like use enzymes to chop up different pieces of DNA and ligases to reattach them. And then you can take that DNA and inject it into a cell. You can electroporate it into a cell, so using electricity to jam it into a cell. You can do all sorts of different tricks to get DNA into a cell. Yeah, making it genetically would be the most broad and correct answer that I could give. Okay, yeah, so that first construct is the normal prion protein attached to a flag tag. And then we have flag CPRP. So this is only the cytoplasmic side of the prion protein. So this entire middle region, for the rest of those constructs, that entire middle region that goes into the membrane and sticks outside of the cell, that's all been deleted. So we're only keeping the part of the protein that sticks within the cell. Then we have the prion protein that's missing this highly conserved modifiable site. So this GPI site, for some reason, evolution decided we're going to keep that part, and we're going to keep that part because who knows why. But for some reason, this part is really well conserved, and we talked about conservation at the beginning of the stream, right? The more conserved a protein is, the more it implies that it has a useful function. And so this construct takes the prion protein and snips off the little bit that sticks into the cytoplasm. Here, in this construct, we swapped the prion's GPI site for a different protein's GPI site. So this is asking the question, is this interaction mediated just by the general presence of this GPI region of a protein? So that's, I think, a very fun and interesting control. Here you chewed off the end of the GPI site, here you chewed off the middle of the GPI site, and then here you chewed off the beginning of the GPI site, right? GPI site highlighted in red here. So if you don't quite understand all these constructs, don't worry, this was just an overview and I will refer and define them again as we need them. But just so you know, as we're heading into these next blots, we are removing bits and pieces, mutating bits and pieces of the prion protein to try to get at what part of this prion protein is mediating this interaction. Oh yeah, and then the flag tag is here too. Okay, so here is where you are going to participate, chat, because this is the part where you will use my guide on co-IPs to try to tell me what the conclusion of these blots are. So we are purifying the full-length CBL protein. That's this guy up here. And we're mixing it with the full-length prion protein. So that's what went into this Western blot. And then here are your fishing rods. Your fishing rod this is the CBL fishing rod. That's for some reason they call it input. Here we have the control fishing rod, and then here we have the prion fishing rod. So chat, you are ready for a challenge, right? Take a guess. What do we conclude here? What? HA tag. This is checking for binding to IGF. So I should have mentioned that on the thing. This is IGF. Right, so we got a tripartite IGF, a tripartite system like this, and I'm gonna give chat the is this IGF? It might be no, it's CBL. It's CBL. It's not IGF. I take it back. Sorry, there are 100 slides. I'm gonna keep on using that as a crutch, but there are a lot of slides. So this is HA is a tag that they put on to the CBL region. So that's why this is called input. I'm guessing that's probably why they called it input instead of CBL. But yeah, so fishing rod and then all of these bands are referring to the protein destroyer CBL. So what is the conclusion here? CBL. What is this conclusion here? I believe in you, chat. When you mix the full-length prion protein and the full-length CBL, and then you go fishing with these 3 antibodies, what do you get? Clockwork says CBL only detected when you use the Prion fishing rod. That's partially correct. Definitely detected when you use the Prion fishing rod, but it's also detected in another condition, right? It's also there in another condition. So I'll repeat it one more time. This is the Prion fishing rod. This is the control fishing rod. This is the CBL fishing rod. And this is the presence of the protein destroyer. Oh, actually, Clockwork, you're right. I'm so sorry. I'm confusing myself. But Clockwork, you are right. So CBL is only detected when you use the Prion Prion antibody, right? So the conclusion, so that's the observation that we can make here, right? That we do have a band when we use the Prion as a fishing rod, we can fish out the full-length protein. This is kind of just letting us know that we can, in fact, fish out, that the lure attaches to the fishing rod. I guess that's the best way to describe this. So this column allows us to know that our lure is attached to our fishing rod. This lets us know that we successfully fished it out using the Prion protein. So our conclusion here is stuff is working and the full-length CBL binds full-length Prion. That's what we should take away here. So this is kind of our warm-up blot, and I want to pause here so that we can get an idea of why this is true. In particular, this band lets us know that we can bind CBL. I wish they had labeled it differently, but whatever. Does that make sense, chat? Here I'll pause and take another poll because you were going to use this logic for the rest of the Western blots. I'm going to go ahead and read chat. They have a lizard model of melanoma called Mr. Frosty, and it has a mutation in a gene called SPINT2. That's adorable. That's interesting. I didn't know that lizards got melanoma. That's very curious. All right, I'm going to take a drink of water. So thirsty. Yeah, so one more time. This is the Prion fishing rod, and this band lets us know that CBL1 was detected. So when we use the Prion fishing rod, that is successful. That is what we should be taking away from here. This antibody successfully binding the protein destroyer. That's the main band that we care about. Yeah, it's a little tricky. We're bouncing between like 75% understanding, which I think is okay for now. It might get more clear as we continue going, but at a 75%, I think I will continue here. Okay. Next up. So, similar format. We are now not using the full-length Prion antibody, but instead we're using the flag tag, right? I said that flag tag would be useful, and it is. This is still fishing for the Prion protein. And the other antibodies are the same. So the only difference between this and this is that we are now only testing, our fishing rod is different, right? So between this slide and this slide, the fishing rod is now just the part of the Prion protein that sticks into the cytoplasm. So what is your conclusion here? I'm sad I need to leave soon because my lunch is nearly over. Oh, I'm so sorry, but I'm very happy that you managed to spend your lunch hour with us. It's really fun to have you all around. I'm also glad to have such an international, actually, I don't even know if you are international, Robert. I kind of assumed because I'm usually streaming at a different time, but I guess lunch is not that far away from where I am right now. Yeah, so what do we think about this this plot here in the USA? Yeah, I figured. I just remembered, like, it's like I usually stream much later in the day, so it feels like 5 p.m. for me. But yeah, I guess if it was 5 p.m., you'd probably still be in the US or like the western half of North America. CBL binds to the cytoplasmic segment of protein, prion protein, confirming interaction is intracellular. Exactly. So I said here that what the conclusion is that full-length CBL binds full-length protein, prion, and the only change here is that we removed the stuff on the outside of the cell. So by removing stuff that binds this, by removing the outside of the cell part of the prion protein, we still get binding to the prion protein, even when the extracellular domain is removed. So this confirms, well, it's not great confirmation, but it is consistent with the idea that the prion protein uses its cytoplasmic domain to bind this protein destroyer. All right. So what's up next? What do we have here? So instead of the full-length prion protein, instead of the prion protein missing the extracellular side, now we have the prion protein missing the whole GPI conserved modifiable area. So this is the whole segment of the prion protein, of the pro-prion protein that sticks into the cytoplasm. What can we conclude here? Now the blot is a little different, right? Now the blot's a little different. Again, conclusion here is that you still have binding when the extracellular domain is removed. What happens when you remove the part of the protein that sticks into the cytoplasm? With no GPI segment, no CBL binding, no binding. Exactly, Wild Max, Clockwork, Robert, you all got it. When you remove that conserved place, you no longer have binding at all. This, you know, makes sense because this region is conserved. So it makes sense that it has some sort of function. Again, it's not guaranteed to have a function in every situation, but when you remove the whole bit, yeah, it doesn't bind anymore. And this is purified protein, right? So we're not even thinking about membrane architecture or anything. This is just purified proteins in a dish. You no longer have that binding. So we can confirm, at least in a dish, we don't know about how it acts at the membrane level, but when we have just the purified protein, this region appears to be very important for binding. So those are sort of the big constructs. Now we have some of the weirder constructs, right? So now we're going to get into this one. Okay, so this is the prion protein, but you swapped that cytoplasmic part of the protein for a different protein's GPI region. What can we conclude here? So removing the entire cytoplasmic part stopped binding. What if we switched that cytoplasmic part for a similar cytoplasmic part? What do we have here? What can we say? Kind of an interesting result here, I would think. What do we have here, chat? What are we looking at? What are we looking at? What can we conclude? Max says still no binding. Exactly. There's still no binding. So even if you took that region, that GPI region, and switched it for a different protein's GPI region, you still don't end up binding. So that you can conclude that this binding is specific for the prion's GPI region. So this is not generalizable, which I think is very interesting. We can go deeper. We can get even finer detail as to how this interaction happens. So now we have, I think this blot is fantastic. This is such an interesting blot. Now we have a blot that has different deletions of the prion protein, specifically these residues. So these are specific deletions. If you've never seen these before, the delta means deleted. So in this first part, you deleted, this first part, 249 to 253. So you are deleting the end of the prion protein. And then here you are deleting the end, the middle of the GPI segment. Sorry, GPI segment. And here you are deleting the beginning of the GPI segment. And so what can we conclude from here? Wildmass asks, why is it interesting, Phi? It's interesting because GPI regions might have a shared function, like some I'm not like a GPI expert. I've actually never heard of them until this Prion paper, but it shows that the function of being a GPI region is not what allows the binding to happen. It's actually specifically the shape of the prion protein at that region that really, really matters. Oh my God, they really went residue by residue. Yeah, it's not quite residue by residue, right? They deleted chunks. It's basically, it's still a lot of work, but it's not like an alanine screen, which is like huge. Yeah, what can we conclude here? Yeah, clockwork. So deleting 4 residues above 249 doesn't seem to hurt binding. Hold on, give me a second. Deleting the last 4 residues, because 249, that's the end of the GPI region. So 249 to 253, this, right? You still get this flag binding. This is the prion protein being able to bind. This is the fishing rod for the prion protein being able to bind CBL. And so when you don't have that region, you don't get the presence of CBL anymore. Does that make sense? So this allows us to tell exactly where in the prion protein is binding this protein destroyer. And again, that's going to be really important for medicine coming up. So I will take a pause just to take another. I know there are a lot of polls today, but I really want to make sure that we have an appreciation for the amount of work that went into this. Region of protein understood? Question mark. Yeah, really, really elegant, beautiful little experiment here, allowing us to pinpoint exactly where the prion protein is binding the protein destroyer. When you delete just the very tip of the GPI region, you still get the fishing rod being able to fish out the CBL protein destroyer protein. When you remove the middle, you have no more binding. When you remove the beginning, you also no longer have that binding. I wonder how they came down to those specific delta ranges. I don't actually know. I don't know if they had a justification. They might have just felt like doing 3 different regions. You could probably go even deeper into it, but that would be a lot more work. If I had to guess, it's probably because doing 3 sections is a lot less work than doing every individual amino acid. All right. Cool. It feels like this stuff should be calculatable as opposed to require manual experimentation. I mean, you have protein folding models like AlphaFold, but at the end of the day, if you're going to make medicine, you should confirm it with your own eyes. Which means peptides or small molecule design dweebs now have specific geometric target for development of treatment. Exactly. This is why I wanted to make sure that we appreciate the science that goes behind medicine development, because now, not only do we know that these proteins bind, we actually know exactly where these proteins bind. So if we design something that attacks this exact location, we have potentially a treatment for cancer. Very, very cool. So you need amino acids 239 to 248 for prion to bind CBL. So that's just the inverse of what Clockwork said, both true. Okay, so there's a ton of blots in this paper. I'm not going to go through all of them. I already went through so many, but we are already like 80 plus slides deep into the presentation. So we're just going to ignore those and say that they did do more of it in order to determine the region on CBL that binds prion. So they did the inverse with CBL instead of the prion protein. So here's a good point to recap for those of you who may have joined us later, the results so far. So the first thing that we talked about is that patients with melanoma have elevated prion levels in their tumors. Melanoma cell lines without prion protein are bad at wound healing and have more metastatic tumors. This suggests that prion protein is involved in metastasis of melanomas. The prion protein binds a protein that destroys proteins. That's CBL. That's the protein that we were just talking about. This is a protein that destroys things. And together they bind a third protein, IGF-1R, which is a growth factor receptor that could contribute to cancer. Specifically, we even know the exact location on the prion protein that is required for this interaction. So we have a lot of mechanistic detail already, but it's still not a satisfying story. We know where they bind, and now some of those earlier questions about, well, how does that lead to cancer? Now we can answer those. So here is the hypothesis. The prion protein is an adapter that brings CBL, this protein destroyer, together with IGF-R1 to destroy IGF-R1 and cause cancer. So this is a cartoon of what the authors think is happening here. It just hit me how cool this is and how helpful it could be. I thought it was really good, but I'm realizing that it's way more potential than what I thought. And that's, I think, the feeling that I get when I read about molecular biology. The application of this research is very powerful. And I think it's very powerful because it is molecular in nature, because we have such fine, precise control of what we can do with biology at these days. And learning more about how things work can really open up the future for drug development. So we believe, we think, we know that this is happening. But we still have to prove that this is actually happening, right? It's bringing these proteins together, but is CBL1 actually destroying IGF-R1, and is it dependent on the prion protein? Of course, we have more blots, right? But I'll just zoom past these because we got through some of the nastier ones. So these are, of course, more Western blots. We love a good Western blot on this channel. And when we do prion knockouts, we actually find that when you have prion protein, you have a certain amount of this growth receptor. When you knock it out, you actually have more of it. So when you don't have prion protein around, you get more growth receptors. The inverse of that is that when you have more prion protein, then you get less of that receptor. Although they didn't show it here, that is kind of implied here. Okay. Yeah, there was something about confounds that I don't really care about talking about anymore because my voice is getting tired. Actually, question, are Western blots always black, or can you get them in funky colors too? So these are, it depends. Okay, it's okay. Blah, 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 blah. When you display Western blot data, you typically make it black and white no matter what, because the actual value that is measured is the pixel density. So it all gets transformed into black and white, so that I don't know if color matters in that case, because, you know, if you do Western blots, if you do it with fluorescent antibodies, they give off a fluorescent color technically. But when you get that data, it's usually transformed to black and white. Yeah, there are some like RNA probes that can do funny light things, but when you see data, it is, for Western blots at least, it is usually transformed into black and white. Am I misremembering gel electrophoresis, or a lot of blots like mega purple? They're not like mega purple, I would say, but they're like bluish, because they are, they like take up the, what is it? Kumasi stain if you are doing SDS, and for Western blots they don't need a Kumasi stain, but they do. You know, the agarose itself is a little bluish, and when you run when you run protein ladders, they come with blue dye, so I think that kind of looks more blue when you run them. What device is used to witness these blots? Is it seen under a microscope or a super microscope? No, so this is actually pretty low tech. You can do it with an X-ray developer, so something that you can, like a photo developer, essentially. That's the old style of doing it. You can also put it in a box that measures fluorescence if you use fluorescence, or you know, that you can call them you can look them up. They're called gel analyzers. They're these machines that are essentially boxes that can take really good pictures. That's basically what they are. Yeah, exactly. Good questions though. Yes, it's the luminosity that matters. Yeah, I don't technically know because I'm not a good physicist if it's luminosity that is being measured, because technically the thing that gets converted into a number is pixel darkness, which I don't necessarily know is the exact same thing as luminosity. I'm just covering my ass here because I'm not that knowledgeable in optics. Okay, so these other cell lines show the same pattern, right? When you don't have prion protein in this right-hand corner, you have less of, you have more of the IGF receptor. That's something that you can see from the numbers. They did a ratio. So you get more when you don't have the prion protein around, implying that when you do have the prion protein around, it destroys that protein. Okay. So, is IGF-R1 being destroyed as opposed to some genetic regulation thing? That's pretty of a nuanced question, but I kind of want to know, like, this protein is disappearing when there's prion protein involved, right? When prions are in the picture, this protein is not as common. Is that because of the cell isn't making as much of the IGF-R1? Is it because it's being like tossed out of the cell, like just yeeted out of the cell for some reason, or is it actually being destroyed? So here we talk about the proteasome. I think Morju said it was his favorite cell thing, or someone in chat, maybe it was Dreamless. But the proteasome, this is a protein complex that is essentially a garbage disposal for proteins. You can see here in the blue region, there's a lot of stuff happening here. And basically, when you get a ubiquitin tag, you enter the proteasome and get chopped up in the body here. So this is a way for the cell to destroy proteins, which is very, very cool. And so trying to understand the mechanism of degradation, the authors use a couple of different inhibitors to check what the mechanism of this disappearing IGF-1 is. So this is not like the first thing that I would have thought of, to be honest, but it is neat. So let's just go through this data bit by bit, and we can see again more and more blots. This is such a powerful S-tier technology. It'll never go away. It's very simple to do, and you can answer a lot of different questions by setting up your Western blots in different ways. So here we have the vehicle control. So these are regular old cells, regular old cancer cells, and they either have the prion gene or they don't have the prion gene. And you can see that when they don't have the prion gene, that you have roughly double the amount of growth factor receptor, right? So that's what they show here. Hey, MG132, that's what I'm working with too. That's cool. I actually don't know anything about these cell lines. It's cool that you're working with them. When you add a proteasome inhibitor, check out what happens, chat. Usually you get an increase when you remove the prion protein. Now what happens when you add proteasome inhibitor, and what does that mean? Right? Oh, not a cell line. Sorry. Yeah, that's the inhibitor. Sorry. Oh my god. Uncle again with making sure I'm not spreading disinformation or getting my own slides wrong. Yeah, sorry. That's not a cell line. That's the actual drug that's used to inhibit the proteasome. Yeah. They all look the same after a while. But yeah, when you add this proteasome inhibitor MG132, what happens to the amount of prion protein? What happens, chat? When you stop the proteasome from working, what happens to the growth factor receptor in response to the presence or lack of prion protein? I want y'all to take a good look at that blot. Tell me what you see. The numbers below the actual blots, that's the number that you should be comparing. Growth factor increases. In what condition? In what condition? Dunkel says it's not affected by the amount of prion anymore. Exactly. So by adding the proteasome inhibitor, you no longer have an effect on the growth factor receptor, right? Whether or not you have the prion protein or not have the prion protein doesn't really matter anymore. So the proteasome stops this IGF from actually working. What's interesting to note is that this proteasome inhibitor causes a baseline increase of prion protein, right? This is 2.3 compared to 1 over here if we're comparing groups, which is notable. That might just be because the prion protein is also no longer being destroyed, but what's important is to compare these two groups. It's no longer affecting, or it's no longer responding to the presence of prion protein. So you're jamming up the way that the prion protein is working. We also see that this is not the case for these other inhibitors, right? The phagosome inhibitor, 3MA, you still have that same relationship where when you knock out prion protein, you get more IGF. So nothing about this really changes. The phagosome, if you watched my video on tuberculosis, is like, instead of a garbage disposal for the cell, the phagosome is more like a little stomach, I guess, like these little vesicles that contain enzymes and acids and stuff that chew up protein, a different way for proteins to be destroyed. So if you inhibit the phagosome, you still see that same relationship. Yes, prion, no prion, you get that same change. Similarly, the lysosome, that's the other half of the phagolysosome, like the some components, they don't appear to change the relationship of prion protein and the growth factor receptor. Thank you for the sub, Gecko Dude. So this is really good evidence to show that binding prion protein causes a proteasome-dependent degradation of this growth factor receptor. And so this implies that because you need the proteasome to do this, that the ubiquination coming from the protein destroyer CCBL1 is what's doing this thing. And so, Longtail, if you were working with a proteasome inhibitor, I wonder if you've also worked with this antibody. So this is K48. This is a ubiquitin antibody. So this is actually a pretty unique Western blot here. A couple of you were pointing out how messy the Prion blot was. Well, this is like omega messy, right? So this is an antibody against the ubiquitin tag. And part of why it's called ubiquitin is because it is so ubiquitous in the cell. So you are going to get this protein binding to a ton of different stuff. So it's not a surprise to see that these are very messy blots coming off of this antibody. So what this represents is that when you have ubiquination, you have protein destruction, and the protein destruction is represented by smears. So instead of all the proteins coalescing in like very tight bands, you get this like general shearing of things. This represents population of proteins that are getting cut up at any given point in time. So you get just like a mess. From what I understand how this antibody works, I've never worked with this antibody before. And you see that when you don't have the prion antibody, when you don't have the production of the prion protein, you get a lot of that messy ubiquination here. So this is, again, another piece of evidence that suggests that this tripartite trimer is destroying this growth factor receptor, potentially impacting cancer development. So we can go back, now we're going to scope back out to the in vivo stuff, right? So in vitro, you were playing in a dish a lot. Now we're going to see if what we learned in the dish applies to stuff that is closer to a real person. So brown here in this pathology thing is the presence of protein. And we can see that in the healthy cell, not a lot of prion protein. We can also see that there is a whole bunch of this receptor in the healthy cell. When you have melanoma down here, a lot of prion protein, and then the inverse relationship, not a lot of growth factor receptor. So this again is another thing that is consistent with this story so far, that prions are destroying this growth factor receptor in real humans. This is a biopsy here. We're doing human experiments. This is not really an experiment. This is just because an experiment requires testing, control groups and hypotheses, et cetera, et cetera. This is just taking patient biopsies and seeing. It's observational, more likely. Yeah. Good to clear that up. Isolated tissue, exactly. These are biopsies. So again, another piece of evidence, prion healthy, well, quote unquote healthy, is a little bit of a variant of the prion protein, but it is a normal variant of the prion protein, is inversely correlated with this growth factor receptor. So what is IGFR1 doing? So again, we talked about this thing being a known regulator of autophagy or internal cell digestion. So this is a receptor that sets off the stress response that allows cells to recycle what they currently have for the production of energy and to do other things. So what I found interesting in my brief perusal of the autophagy literature is autophagy can either increase or decrease with regards to cancer depending on the context. I think in most cancers it's upregulated, but in this specific case it seems to be downregulated, which I think is pretty unusual. Or did I get that mixed up? Hold on. So many things in biology go up and down, so maybe ignore that last statement. I kept it vague because it is confusing. IGFR1 and autophagy, these things can be dysregulated in cancer, so they can be inappropriately high or inappropriately low, and that can lead to cancer. I'll just put it that way so it's not as confusing. So in cells that lack prion protein, you have less autophagy happening, right? So here, the relationship between IGFR1 and autophagy, I think, can be kind of confusing. I'm about to lose it. I'm getting myself mixed up here, but the data don't lie, so we just look at the data, right? So M2, this is a marker for autophagy. We see in these fluorescent cells that when you do have the prion protein, you get more of that autophagy happening. When you don't have the prion protein, you get less of it happening. Yeah. Yeah. Okay, yeah. Never mind. That makes sense. Because we suspect that if you have, if you have more autophagy, that can lead to more cancer. IGF-1 in this situation is decreasing autophagy. Yes, it's a mess. You do that all the time in biology. Up, down, up, down, up, down. So adding prion back into prionless cells destroys IGFR1 and increases autophagy. So this is a rescue experiment. This is just double checking things and making sure that the relationship holds from an instant point of view instead of a developmental point of view. So when you add prion protein back into the system, what you see is that you are able to destroy IGFR1 and increase your autophagy. So here in this first column, you have nothing added. You have a regular amount of IGFR1. Here you have a fragment of the prion protein, and you are back to destroying the growth factor receptor. And in fact, this particular region, this is the region we know the prion protein needs to bind the protein destroyer. So that makes a lot of sense. And then these two conditions, these are the parts of the prion protein that actually don't really matter for binding. And so that actually is reflected in the fact that they don't really change the IGFR1 levels. And then when you add the full length, you get destruction of that IGFR1 again. So we have a model of prion's involvement with skin cancer. So you have prion accumulation at the cell membrane. That prion protein binds this protein destroyer CCBL. This protein destroyer destroys IGFR1, which decreases autophagy. Increases autophagy. Let me make that edit right there. I think I got that wrong. Should be increases autophagy. Sorry guys, this is so confusing. It's confusing even for me, the way that these interactions happen. Let me just take a second to double check, making sure that that is true. It should be, I believe it, yeah, it should be increased autophagy. Let's correct that. And I can't edit this. That's unfortunate. Okay, so just know for the VOD watchers that that's meant to say increased autophagy. And I can't edit it because my license for PowerPoint has expired and I'm having trouble renewing it. But we continue. So how do we test this? So if we break up the interaction, we can see if that stops metastasis, right? So we were talking about how we could potentially design a medicine based off of this interaction. So this is now, I think, a very fun point in the presentation where I get to mine your ability to come up with a way to design a medicine to disrupt this interaction. And I'll give you a hint, chat. We already know from this paper a protein that fits exactly into that pocket. And if you have an answer, give it actually a couple seconds. I'm going to go grab myself a bit more hydration, and I will be right back. But for now, think about... So let me just change this. Think about how you could design a molecule that fits right into that nook that could break up this trimer. So we'll do thinking time. I'm taking a water break. I will be right back. I'll be right back, right? We already know that there is a protein that fits right inside that nook. And think about what that protein could be. I'm going to get water. All right, back with some water. What did we all come up with? We already know that there is a protein out there that binds specifically in that region. And it's a bit of a trick question. And I want to allow that question to hang for a bit so that when I reveal to you what the answer is, it's going to be all that more mind-blowing. So we already heard about a protein that fits in there. Anyone else want to take a guess besides Leo? Anyone else want to take a guess? The answer was in us all along. The friends we made along the way. It was right in front of us the entire time. All right. How about those 8 or so amino acids in the prion protein that we identified as being important for binding? This wasn't the flag, was it? Ubiquitin? Yeah. Yeah, Leo. It is included in the flag story because that's where we learned about it. It's exactly as Lunkil said. We already know. We identified with those Western blots, with those co-IPs, the exact 8 amino acids that if we stitched them together, binds in this exact spot. Which means we have a medicine angle. We have an angle, right? We already know. I'll zoom back to those slides. We already know from... Where was it? This here. Right? This is back when we were doing those co-IPs. We learned that this region between 239 to 248 is really important. These two blots. When you remove these sequences from the prion protein, you no longer bind the protein destroyer. So we know that this region is really important for this binding. So we have already built into this research an angle of attack. And I think that's just incredible, right? That's amazing. That's so clever. I would have never have thought of it. If you forced me to, maybe I could come up with it. But to me, yeah, that's pretty incredible. So we already know from before in this paper that the 8 or so amino acids that we identified as crucial for this binding to happen should hypothetically fit into that pocket. Now, of course, we have to test it. It's not a given because, it can interact with all sorts of other things, get stuck in the membrane, who knows. But we have a very promising candidate that binds exactly in the correct location. So if we break up this trimer, if we try to break up this trimer using that peptide, not only do we have a way to confirm-ish the mechanism by which this works, but also an angle for an actual cancer treatment coming out. So yeah, exactly. Remember, you need the amino acids 239 to 248 to bind this protein destroyer. So here's the experiment. this is the peptide 239 PVILLISFLI 248, proline, valine, isoleucine. Oh, I can never remember. Leucine, yeah, leucine, isoleucine, serine, phenylalanine, leucine, isoleucine. This sequence of peptides shown here, the authors are going to use this to try to jam this interaction here, because the authors know that this sequence of amino acids should perfectly fit in between here and break this interaction up. And if we can break this interaction up, we can save this protein and prevent cancer. That is their goal here. But before we get to that, do we have any questions in chat? Because this is a really, really important moment for developing your sense of experimental design. It's a moment of like, work smarter, not harder. And I want to sit on this idea for a hot second, just to appreciate making good work with the data that were already generated. How do we get individual proteins into cells anyway? Do cells just kind of allow in all proteins, or do we need to infect them like a virus does? That's an excellent question, Leo. So you can do it with a virus. Most famously, you can do it with a virus that a certain YouTuber did to remove their lactose intolerance. That is something that you could try. It can work. Whether or not it does has to be rigorously tested, of course. But there are all sorts of other ways that you can introduce proteins to cells in the body, like cell-penetrating peptides that you can borrow from the HIV protein, the HIV virus. So the HIV virus makes a protein where if you attach that protein to a protein, that protein allows that protein to sneak through the membrane, which is very good. You can do it through things like lipid nanoparticles. So lipid nanoparticles can be uptaken by cells. So that's a particular option. Even better if you have a way to conjugate a cell-specific antibody to a lipid nanoparticle, that could do it too. So there are tools that biological engineering has created to allow you to get individual proteins in the cell. Now, if you were to do it the virus way, the way that the YouTuber who will not be named did it, that involves a little more genetic engineering that many people are comfortable with. And so by doing something like cell-penetrating peptide or lipid nanoparticle, the treatment is transient, right? You put it in, you take it as a treatment, but it's not permanent. But it does involve not introducing DNA that you do not have into your body. You know, I make it seem like I'm crapping on the idea of gene therapy, but I'm really not. It's just that the usage of gene therapy has to be very targeted and very carefully thought out and very responsible. Because if you've ever taken an engineering class, you should also take an ethics class. So when you are designing your treatments, make sure that you are following good practice in that way. All right. Lipid nanoparticle sounds like 1998 sci-fi. And then wait till you have gold lipid nanoparticles, which are now being used in skincare, funnily enough. Which I've been thinking about doing a skincare stream. I might take a poll at the end of the presentation to gauge interest, if you would be interested in the science of skincare. I think that'd be kind of fun. Okay, so very, very cool experiment trying to break up this interaction, trying to see if it stops metastasis. So hypothetically, we could sever this interaction by slipping in this region of the prion peptide. Yeah, so this peptide can compete for binding, so this is a competitive interaction. If you watched my stream with Clockwork, you would have heard about competition. So if you want to design treatments, you can design things that are meant to bind to each other in a way that disrupts existing bindings. So that is one way that medicine can happen. If I stream about that pill that claims to be sunscreen, why don't you drop that in the Discord? I'll check it out. So here we have the co-IP again. Of course, the co-IP Western blot, very, very powerful. So here you have the setup here. So you have your bead, you have your Prion antibody fishing rod, you have your Prion lure, and then you have a mixture of the peptide, this peptide that we previously identified and the protein destroyer. And here, along this axis, we have increasing amounts of this peptide. So that's what this P10 stands for. So when you have no peptide, you have a good chunk of CBL. You have some of the peptide, you see that band getting fainter. And then you have a lot of the peptide, you see that band almost entirely gone, right? So we can confirm that in a dish, you can break up this interaction with this peptide, which is very, very interesting. So does this have implications for IGF and autophagy? And the answer is yes, right? So LC3-2 is a marker for autophagy or cell digestion, a pathway implicated in metastasis. So you can see that as you add the peptide, that's what this P10 is, you save IGF-1, right? You have a baseline of 1. After you add that peptide, you have more IGF-1. More IGF-1, you have increased... you have decreased autophagy, right? You have decreased autophagy. So this is the bar we're looking at, and then the ratio is 0.5. So you are decreasing the amount of autophagy, which should help with controlling this cancer. So let's go back and look at wound closure and relative invasion to see how metastasizing these cultures are. Excuse me, after you've added peptide. And so when you add the P10 peptide, this peptide that we previously identified, you can see that there's a pretty big drop in wound closure, right? So these cells are not as good at healing themselves, which is actually a good thing when we're talking about trying to fight cancer, right? And they're also less invasive. So this isn't as dramatic as not having the prion protein as a whole, but it is meaningful, right? Like, this is pretty tight. The spread is pretty tight. It's pretty consistently not as good at being cancer in a dish. What about an actual tumor? So this is now a graft. So they grafted a tumor onto the lungs of these mice. So this allows these tumors to exist in a biologically more relevant context. And if you count the number of tumors, you can see that the mice that have had the melanomas grafted have a very high level of metastasized tumors. But when you give them the peptide treatment, they fare a lot better, right? A lot better. Nowhere near as many metastasized tumors. And in addition, you have a pretty big drop in tumor. Well, I guess it's only 10% between the two, but it is still a pretty noticeable difference in tumor volume, tumor area. Looks like it's a way more pronounced drop. What do you mean by pronounced? I seem to remember that if you remove the prion protein as a whole, I believe that figure at the beginning had relative invasion like all the way down here. Although you might be referring to something else. But yeah, so this is kind of like the in vivo verification that we wanted at the beginning, right? But in order to justify, let's be real, hurting a mouse, right? Like this mouse had, people gave it cancer. But in order to justify doing that, you have to really make sure that the payoff that you get is worth it. And, say whatever you want about animals and the use of research. If you want to cure diseases, you have to make sure that you test it on something that is actually alive, right? So here we can actually see that this peptide is in fact doing something, and that's very, very exciting, even in an in vivo system. Now, this is, I think, yeah, this is melanoma grafted mice. I wonder if there is a model of mice that don't have to be grafted. That might be a little more physiological because, of course, grafting a tumor is not exactly the most normal way to have a tumor. Very interesting in that the peptide works so well when it's just straight copied from the protein, not even optimized for binding affinity or anything like that. Yeah, like it maybe with optimization it works even better, right? You've only got 10 amino acids to play with, so like maybe you could really make this a great disruptor of that interaction. Who knows? So we are miraculously at the end of this stream, so I will give you all a summary. Prion, the special version of the prion protein that sticks out in the cell cytoplasm, is elevated within melanoma. And we have reason to believe that it may be destroying an autophagy regulator, IGFR1. And then in melanoma, this results in, I always get this confused. This ends up with increased autophagy, which leads to metastasis. If you decrease the prion protein by genetically removing it, or if you add the cytoplasmic domain of the prion protein, it stops IGFR1 from being destroyed and may actually stop metastasis. So from like the amount of progress this paper made is unfathomable, right? Remember, if you were here at the beginning, this paper started with a data mine, just being like, what genes are upregulated in metastatic melanoma, and we got from what genes are involved all the way to a potential treatment is absolutely bonkers. So how robust is this model? We'll just take a quick recap on the evidence that we had along the way. So proprion levels rise. We have the knowledge that proprion is in fact elevated in metastatic melanomas, and co-IPs and Western blots gave us a lot of information about how these proteins work on each other. And then we had more Western blots that told us about autophagy markers. And then the link to metastasis, I felt like, is not that clear, although we do know that disrupting this interaction, well, let me pull back, that adding a peptide that we think disrupts this interaction, or that we did show does it in a dish, is leading to a better cancer phenotype. And I want to probe by asking, and this is like really graduate level thinkies here, right? This is something that I think is a very hard thing to try to parse, but if you can successfully pick up what I'm putting down, I think that would be incredible, right? So we have this peptide. We think that when we add it to this system, that it breaks up this interaction. We have evidence that it does. Do we know with 100% certainty that the peptide is stopping cancer by breaking this interaction up? We know that it does break this interaction up. We know that the peptide does help with cancer in a dish. But can we connect that action of the peptide directly to the disruption of this complex, to the decrease in metastasis? Did we prove that in this paper, yes or no? Give that a little bit of time to think while I hydrate. And I think if you've been following my channel for a while, you kind of already can tell when I ask when I ask a leading question like this, it's there's usually something to it. There's usually kind of a trick involved somewhere. I'm wondering if we can sniff that out. Can we connect directly the peptide to this breaking up of the interaction all the way to stopping metastasis in melanoma? Are all of those chains unbreakable? Yeah, kind of hard. Kind of a hard question to answer. Now that we've kind of sat with that question, I'll go ahead and talk through my thoughts. The answer that I have would be no. We have a lot of reason to believe that it is true, but the links and logic are not airtight. And I don't think that the authors meant for it to be airtight. And this is kind of a bit of the nuance that I want to get across with regards to science communication, is that like the reason why science is wrong a lot of the time, not even a lot of the time, but the reason why things change in science is because we're always updating our models, our current understanding. And there's a little bit of wiggle room for the peptide to be doing something else entirely, right? Maybe this peptide is doing something that has nothing to do with this trimer, something completely different, and that's the actual thing that's stopping melanoma. And maybe the disruption of this complex is just a side effect, right? Say it with me. Correlation is not causation. We do not have causative proof that this peptide works in the way that we think that it does, but the evidence is pretty good. It's pretty good, right? It's not terrible, it's not bad, but the link isn't airtight, and that's not a criticism of the authors, that's just an observation and an acknowledgement that there is more work to be done, right? Science is imperfect, that's why science still exists. If you had the perfect experiment that left no questions unanswered, science would be over, right? There are other ways to more conclusively say that this is how it works. Messing with CBL chaperone proteins that affect final folding instead of direct binding. Such small peptides can have many different places to stick to. Yeah, exactly. Like, who knows what this thing is doing? It looks like it could be stopping metastasis through this mechanism. It could be. In fact, I would even wager to say that it's very probable, but I wouldn't bet $1 million on it, right? Like, it could still be doing something funny. And the way that you would test that is you have the interface by which CBCL1 binds to the prion protein, right? Like, you know that it binds here. And what you can do is you can mutate the peptide so that it no longer binds CBL. And if you do that, and you introduce the peptide and it no longer works, that is much better. It's not perfect, right? This is a very hard question to answer perfectly. But if they had taken the peptide, and if they had made, you know, a couple of different changes, maybe 1 or 2 different changes to decrease its binding affinity for the proposed target, and try to do that as a control, that gets us a lot closer to being that much more confident that this is the mechanism, right? Again, that is to say, it could still be doing something else that relies on that exact protein sequence, but it's impossible to know it, you know, just from this amount of data. Yeah. I think that's all I have for this story. I will leave you with some other prion protein papers that I came across while doing research for this journal club. So apparently the prion is like a known thing. Like, this is from 2010. You know, that's quite a bit ago. It's like known that the prion can play a role in human cancers, which is crazy to me. I had no idea. Even glioblastoma, which I like, you know, because I did the glioblastoma stream, I have no idea how I didn't catch this before. But apparently, it's doing something there too. And then, of course, this is a big review of all the different ways that the prion protein can function in different types of cancer. So if you are curious and you want to learn more, feel free to join the Discord, first of all, but also you can go ahead and just Google these titles. I forgot to put them in the description, but you have the names up here. And yeah, I think that is my last slide. Yeah, that is in fact my last slide. So let me go ahead and end this stream back on my title slide. I'll go ahead and hide this for a sec so I don't flash everybody by scrolling super quick, to end with my announcement here. So yeah, the Prion video, Come Hell or High Water, will be done by Wednesday, barring any personal tragedy, I suppose, that will prevent me from hitting the publish button. But please look forward to watching that. I worked really hard. Also, Clockwork did a huge chunk of the work animating a very interesting aspect of the influenza story, and I'm very, very excited to see what you all think of that. So yeah, with that being said, that's all, and I will spend a couple minutes hanging around to answer some questions, but if not, that's a freaking stream. That's a freaking stream. Go ahead, hide this. I'll go big mode. Yeah, so what did y'all think of the paper? Did you enjoy it? Was it hard to understand? Was it accessible? Did it leave you with any other burning questions or any ideas of your own? I'm kind of curious, those of you who have stuck around with me to this point in the stream. What do you think about prions, cancer, etc., etc.? Or I'll also take general biology questions for the last little bit here as well. What do y'all think? Go ahead and hydrate. Man, talking is so tiring. The weather has gotten a lot nicer, which means I have to start remembering to wear sunscreen again, which again is really important, one, to prevent melanoma, but also there's been this like running, not gag because it's just reality, but when I go to my class's graduations, I've been mistaken for an undergrad every single time, and it dawned on me, I think last year, that there will come a day where That will no longer be the case, where I will no longer be mistaken for an undergraduate at my school's graduation. Um, Turns out that might never be the case because, as people in my Discord already know, I will be ending my job there. And so I can perhaps say that I've been mistaken as an undergraduate every single year I went to graduation. And I have begun to take a lot better care of my skin because I think the day that that stops happening is tragic. I tried doing a co-IP myself and it didn't really work well because of horrible unspecific background signals. Yeah, that's why the number of co-IPs they did, very impressive. I also did co-IPs when I was in graduate school and they were hard and bad and I had a low abundance protein and it sucked. So this paper made me pretty envious of their co-IPs. Me too, Lunkel, me too. Canada? No, I am not from Canada. Well, I do not reside in Canada, I'll say. Yeah. So the day that I get called out for my current age is a day that will go down in infamy. So I highly recommend, especially, you know, if you are young, please take care of your skin. It's worth the investment, and don't do like the typical guy thing that I was guilty of. You know, when I was young, I tried like one skincare product, it didn't work, and then I never tried any again until Toxo introduced me to her regimen. And now I, for the first time in like many years, I actually have skin that I don't have disdain for. So if you're not taking care of your skin, try a couple of products out. You might be surprised, right? If you don't get it right the first time, it's not a reason to quit trying to take care of your skin. Although I will say that like skincare science is very tough to evaluate because, you know, if I'm going to be applying medicine to my face, I kind of want to know how they work, and a lot of those papers are unfortunately paywalled, so it's kind of hard to, or not paywalled, but rather not on kind of the information market, right? Because a lot of those are probably proprietary. That being said, I will say skincare stream, question mark. Is there an appetite for skincare? And remember, like, I'm not asking if it would be fun to do a skincare stream. I'm sure it would be, but it's more like on the priority list of things that you want to hear about. Is skincare the upper half of interesting ideas or on the bottom half of interesting ideas? I think that is a better way to phrase that question. I think this is one of the first few papers I saw but did not read that prompted me to try and work with prions and tumors for my undergraduate thesis. So it was super interesting watching the stream. Yeah, I'm glad. I will say it is not easy to work with prions. I will warn you. They are, you know, dangerous beyond imagination, right? I highly doubt responsible adults will let you work with purified prion protein. And if they do, I don't know that they're BSL-4, but they are very difficult to work with. Not to discourage you from wanting to work with prions in the future, but I'm just saying, like, keep your expectations realistic. And also, this protein is a MFer to actually work with, because any aggregating protein is a nightmare to purify. So that being said, there are other ways to study and research prions that don't necessarily involve purifying them. But just know that, like, if you are going to fall in love with prion science, make sure you are prepared for the heartbreak when your experiments don't work, because they are very tough. I never got ads on your stream before, but this time I got them at completely random points throughout the stream in the middle of sentences, which was kind of annoying. Yeah, the OBS is supposed to, or Stream Elements is supposed to put it at natural breakpoints, but they don't even let me know when the ads are running, which is kind of annoying. I'm experimenting with monetizing streams with ads for a little bit. I might just remove them because I value, because streaming is not really a moneymaker for me. I mean, I appreciate all of you who are members, right? Your support means a lot. But when I am entering my newfound era of trying to put food in my mouth based off of YouTube, streaming as of right now is not that big of a money generator. So I'm just experimenting with ads for now to see if it might be something that I could look into in the future. Or I could push membership more heavily and then not do ads at all. Yeah, I'm sorry that they were randomly in the stream. That is kind of annoying on my end too, because yeah, I have no idea when those are going off from my OBS. I'll probably only do one more ad containing stream just so that I have an initial data set to compare monetized streams, monetized with ads involved versus without involved, so I can calculate kind of if it would be worth it to keep ads on, or if there's anything I can do to like mitigate the effect of ads on the viewing experience. I would have to do a little more research to see if OBS will allow me to do that. All right. Yeah, so I'm really shocked. 100% of poll responders said that a skincare stream would be upper half of interesting topics to cover. So I mean, I might have to put a little more thought into what I could get. Now, I might not do it because a lot of that research is in fact paywalled by skincare companies. If any of them want to sponsor me and give me some of their research, that would be really cool. Right? Like, I could not advertise a skincare product on the basis of its efficacy, but I could advertise a mechanism of action if I find that to be cool, not from like a doctor or a dermatologist point of view, but just like, you know, did you do science, compelling science in a dish? I'm qualified to talk about compelling science in a dish. That'd be neat. All right. Yeah. I am tired. But I had a lot of fun. I always have a lot of fun talking about science to you guys, and I'm always very pleased to have you all show up. Exciting video release on Wednesday, and exciting news for the future. Look forward to the V Theorem 1 year anniversary in May at some point, probably. Toll-like receptors would be a nice topic to look forward to. I have a video idea in mind talking about the life and death of a neutrophil, and we will be talking about TLRs in that video that will come out, I hope, in October or something like that. Is there an option to manually run ads at some opportune moments? I think that there is. Oh, you know what? I'm going to try that right now. Let me try that right now. Give me a sec. Okay, so okay, I just inserted an ad. So let me know if that ad played for you all. That might be the way going forward. I wonder how much of the stream was kneecapped by containing the ads. That works. Great. Did it work for everybody? Did everybody get an ad? It's kind of weird how this works. I don't know if you get the ad if you are an ad blocker enjoyer though, but did you get an ad? Let me just test that now, because if I can just do that and it works, then I guess that just happens from now on. I didn't get an ad. I'm on phone. I don't get it. I don't like, and you don't. I don't think is this a problem on Twitch? Because on Twitch, I think everybody gets the ad at the same time. This happened in the last stream too. 80% said no. Most people did not get an ad. I don't know how YouTube does ads, man. I don't know what's happening. I have no idea. What the heck? Bizarre. Yeah, I also didn't get the ads last stream when other people did. Okay, I'll add ads manually going forward. Now I'm on premium. We've had premium members get ads too, which is really bizarre. And I don't love that, because if you're paying for premium, like, what the heck? But in the future, I have found the insert ad button, so that is how I will be handling ads from now on. You're right. I think maybe there's a chance that like during when I'm asking questions to chat, if people get ads, then that kind of ruins the whole like interaction bit. So I will keep that in mind, and I will run ads manually from now on. All right, thank you all for showing up. Lovely to chat with you all. I will see you all at some point, who knows, but hopefully a lot more often in the future as we enter a new brave Phi territory. But until then, bye y'all. Bye bye. Bye bye.